I'd like to thank the organizers for inviting me. Today I'm going to talk about determining genetic susceptibility to food carcinogens using Saccharomyces cerevisiae budding yeast, and I think I'm the only yeast person working or speaking at this meeting. This work was done at the Colleges of Nanoscale Sciences and Engineering at the State University of New York Polytechnic Institute in collaboration with Patricia Egner, who's at the Johns Hopkins University. So I'm having.
It's not working?
I'm having a little issue here. It's not.
Let me try.
Sorry.
Yeah. No. It's
No.
here. Should be. No, yeah. Yeah. I tried to.
What happened?
Whole presentation. Yeah.
You want to go to full screen, right?
Yeah. No, but it's not working.
It's not doing it. Okay. Give me a second. It was just there. What happened?
I don't know.
Let's go ahead and turn it on. Let's go ahead and stop it.
Up to you.
Yeah, let's go ahead and stop that real quick. And then reopen it. This is him, right?
Yes.
Okay. Yeah, there it is.
Okay.
Oh, but I see what you're saying. Okay.
Okay, I think I got it.
You
now.
got it? Sure.
Okay, sorry for that. Okay, so our hypothesis is whereas a small fraction of cancer is caused by high penetrant genes, a majority of sporadic cancer is caused by a combination of interaction between low penetrant gene and environment interactions. And so the outline of my talk, basically I'm going to talk a little bit about diet and cancer. Now we'll talk about sources of food carcinogens, talk about activation of these food carcinogens by P450s and P450 and N-acetyl transferases, talk about characterization of P450 polymorphisms, some of our results with high throughput screening for carcinogen resistance, and then terminate with pathways for activation of colon cancer carcinogens. So I think many of you are familiar with literature that diet is linked to cancer. And some of these studies are actually quite old. They date from the 1970s. And basically what's been observed is that immigrants to the United States switching from a vegetarian diet and adopting a meat-based diet lower in dietary fiber have a higher incidence of colorectal cancer. There have also been studies of those inhabitants of China that have switched more to a Western diet, also having a higher incidence of colorectal cancer. And then there's some specific cases of specific food items. For example, the Chinese in Guangdong province, in which there's a very high incidence of liver cancer, when they switch from a corn-based diet to a rice-based diet, they have a lower incidence of liver cancer. So, what's in food that actually causes cancer? This is steak and corn, which is staple in Midwest and in the South of the United States. And so one thing that's been noticed, especially in corn and in groundnuts, is that aspergillus contamination can occur. And this is especially a problem in subtropical areas of Asia and in Africa, and it's becoming more of a problem in the United States due to drought. And aspergillus forms this very toxic compound known as aflatoxin. Aflatoxin, as many of you know, is the most potent liver carcinogen that we know of. Fortunately, the FDA puts very strict limits on aflatoxin. The limits are 20 parts per billion for humans, However, animals are not so lucky, and periodically there are recalls of dog food and animal feed because of higher levels of aflatoxin. The problem with red meat, as many of you are aware, is that when you cook red meat at very high temperatures, the components, creatine, sugar, and amino acids, that form chemical reactions that form heterocyclic aromatic amines. This was introduced in previous talks by Dr. Anderson. And these heterocyclic aromatic amines are diverse compounds, including IQ, MEIQ, and PHIP. And if you want to know the full names of the acronyms, you can refer to my abstract. So these compounds are not immunogenic per se or genotoxic per se, but they need to be activated, and they're activated by cytochrome P450 enzymes. In the case of aflatoxin, it's activated by a liver-specific CYP1A2, and that forms a very potent epoxide, which can react with protein, RNA, and DNA. When it reacts with DNA, it forms these N7 guanine adducts, which we actually can detect, and this inhibits replication and transcription. Aflatoxin is also activated by cytochrome P450-3A4, which is present in the liver. It's also activated by extrahepatic cytochrome P450, such as CYP1A1, but these form additional metabolites, such as the hydroxylated form of aflatoxin. Heterocyclic aromatic amines are metabolized by cytochrome P450 enzymes, and they form these N-hydroxy derivatives, such as N-hydroxy-IQ, but they also further activated or in an attempt to detoxify them by N-acetyltransferases, including NAT2, which forms this acetoxy-IQ, which is strongly susceptible to heterolytic cleavage, which forms a nitrinium ion, which is very highly reactive and reacts with DNA. So what are the genetic risk factors involved in food carcinogens? So these are alleged genetic risk factors, including polymorphisms in P450 genes. As I mentioned, they're potent DNA damaging agents. When they're activated, polymorphisms in DNA repair or cell metabolism. And then probably the most strongest evidence is polymorphisms in genes encoding phase 2 enzymes, such as NAT2, and the rapid acetylating phenotype of NAT2 is correlated with cancer. So why are we studying yeast? So I think many of you are familiar that yeast is important in making wine and bread. But it's a model eukaryote. Budding yeast also has no P450 enzymes that can activate the carcinogens. And so we used yeast as if it's a test tube for actually introducing different P450 enzymes to make different types of carcinogenic metabolites. In addition, 31% of the yeast genes are orthologous to those that are found in humans, including those that function in DNA repair, replication, and housekeeping function. There was a very important paper that appeared in Science recently in which half of these essential genes can be replaced by human genes and restored viability in yeast. The DNA damage response can be easily profiled. We can use systems biology approaches in terms of looking at transcription, resistance profiling, and proteomics. And we can also look at damage to eukaryotic organelles, such as the mitochondria. So one of the assays that we developed to look at genotoxic effects in yeast is some recombination assays. And this is sort of analogous to the Ames assay, except that we're looking at recombination as opposed to mutation. And I should add that aflatoxin is actually not a potent immunogen in the Ames system. So essentially what we've done here is that we take fragments of His3, which overlap by 300 base pairs. We put these fragments on different non-homologous chromosomes, and by homologous recombination, we can regenerate the wild-type His3 gene, and in so doing, we make a chromosomal translocation. So we can plate these cells on media lacking histidine, and what you see that's sort of analogous to the Ames test is that when you add a chemical agent, you see a ring of induced colonies. And so one reason why we really like this assay is that the spontaneous frequency of recombination is so low, so most of the recombination events that we see are actually induced recombination events. So we actually have minimized our background in this particular assay. So aflatoxin is very strongly mutagenic. We can detect recombination events at levels around 10 micromolar. We can also do growth curves, and what we've seen is that exposing cells that are expressing CYP1A2, these cells are more sensitive to aflatoxin. Aflatoxin most likely induces some breaks to stimulate the homologous recombination because yeast is very good at repairing double-strand breaks by homologous recombination. So the RAD52 gene, which is defective in homologous recombination, shows enhanced sensitivity to aflatoxin. So we've used our system to actually phenotype cytochrome P450 alleles. So these are human alleles. And so one reason why this study is important is because these alleles occur at very low frequencies in the human population. And so it's difficult to do epidemiological studies when you have alleles that are occurring at such a low frequency. But they've been detected in the Chinese, French, and Japanese population. And so one thing that we can do is you can take these the fragments of these genes or the full-length cDNA that contain these alleles and express them in other organisms and see if they will induce mutations. This has been done by one group in which they took fragments, or they took the fragment that lacking the N-terminal sequence, expressed it in E. coli, and what you can see here is that the I386F allele, which maps close to the activation site, is actually a poor inducer of the heterocyclic amine IQ. So we've introduced some of these fragments into yeast, or we've actually introduced the full-length cDNA into yeast, and then we've quantified DNA adducts, toxicity, and DNA repair mutants. And you can also look at DNA damage-induced foci, such as RAD51 foci. So sort of as a proof of principle, what we can see in this particular slide is that when we knock out DNA repair genes, such as RAD4 and RAD51, we essentially eliminate most of DNA repair, and these cells are extremely sensitive to aflatoxin. So we can then introduce these P450 alleles, and again, what we see is that when we introduce the I386F allele, the strain is fairly resistant to aflatoxin. So this is sort of proof of principle that we can phenotype P450 polymorphisms. This is another genotoxic endpoint. These are RAD51 foci. The RAD51 gene has been tagged with yellow fluorescent protein. And again, what you can see is that when we're expressing these particular polymorphic alleles, you can see RAD51 foci indicative of DNA damage, whereas the I386F, you rarely see RAD51 foci due to aflatoxin exposure. So we also wanted to extend these studies to other alleles. One is the cytochrome P450, 451, 1A1 alleles that have been well described in the literature. Unfortunately, the literature contradicts itself in many ways because some studies suggest that these alleles are risk factors in lung, colon, liver, and breast cancer. And so one of the salient questions about these alleles is, are they actually, do they confer higher activation on carcinogenic compounds? So we've expressed both the CYP1A1 I462V allele and T461 N allele in yeast and looked at activation of aflatoxin in addition to benzopyrene diallopoxide, which is a lung carcinogen. So this is sort of an ongoing study, but what you can see in this particular slide is that when we express these alleles, we can still get enhanced recombination frequencies due to aflatoxin exposure, not quite so much in terms of benzopyrene diallopoxide exposure. And we can also measure enzymatic activities. So another suggestion that folks have been looking at is that perhaps some of these CYP1A1 alleles activate hormones somewhat differently. So it's really unclear about why some of these alleles actually are tied to higher frequencies of cancer. But this allows us in terms of looking at how they can activate compounds in yeast. Okay, so just a summary of the P450 allele expression. We can express the full-length CYP1A2 proteins. The DNA damage biomarkers can be used to differentiate aflatoxin activation, and the methodology could be expanded to other CYP1A2 alleles, P450 genes, or other genotoxins. So now I'm going to switch gears a little bit and talk about transcriptional profiling and doing genomics using the yeast system. In this particular study that we did in collaboration with Christian Sinkstad, we looked at the transcriptional profile of yeast after exposure to aflatoxin. Lots of genes are upregulated, some genes are downregulated. As expected, many of the genes in DNA repair are upregulated, including RAD51, which is 8-fold stimulated. Other genes are only upregulated perhaps 2-fold. And so one of the salient questions about these types of studies is what's the functional significance of upregulating or downregulating the expression of genes? And so one way of looking at this is actually to knock out these genes and ask the obvious question, do they actually confer carcinogen resistance? And so the yeast genome is, I think it was mentioned yesterday, there are over 6,000 genes in yeast. We could look at 6,000 survival curves if we wanted to. That's actually a lot of work. But fortunately, investigators at Stanford have marked each deletion with molecular barcodes. So essentially what we do is we use high throughput screening. We pool the entire strains together, and we actually count these barcodes before and after exposure. So sensitive strains are identified by the depletion of these molecular barcodes. Okay, and this is all done by bioinformatics, and so if you have further questions, I don't really have time to go into detail about the methodology, but I'm happy to, willing to answer questions. So the human CYP1A2 has been introduced into 90% of the collection. And what we do is then when we have all this data, obviously you get a lot of data from these types of screens. And so there's freely available software from both the University of Toronto and Stanford University. FunSpec is one of the programs. And so you group these genes into gene ontology groups. And as you can see, many of, this is only a partial list, but many of these genes group into genes involved in DNA damage response. But you also identify other gene ontology groups that wouldn't be obvious, such as those involved in iron transport and sulfate assimilation. So these may be actually quite interesting to look at. And I just grouped them, some of these genes that we've identified into various DNA repair components. I don't want to go into detail about it, but one thing I wanted to point out is that we're also identifying genes which have been indicated to be only meiosis specific. So this is sort of interesting. It's also coming up in other studies in terms of looking at high throughput screens for other toxins. Okay, so in the very last couple of minutes, I wanted to talk about activating food carcinogens such as IQ. There's a sort of an interesting controversy. It hasn't quite been resolved in the literature about how IQ actually gets activated. So one idea is that IQ gets activated by CYP1A2 and NAT2 as I described, but the particular problem is that CYP1A2 and NAT2 are liver specific. And as I mentioned, IQ is a colon carcinogen, not a liver carcinogen. So one idea that's been circulating in the literature is that these compounds are actually glucuronidated, and this gets released into the bile, and then it goes into the colon. And then what happens in the colon? Well, there's microbial flora, and these microbes produce glucuronidases because they want to grab the sugar. When they grab the sugar, they reproduce this nitrinium ion, which is highly reactive, and that causes mutations within the colon. And so the other idea that's coming in the literature is that IQ activation actually occurs by some colon-specific P450s, but these are actually expressed at a very low level in the colon. So we've actually been looking at some of the genotoxic assays, and this particular slide, which had been previously published, basically just shows that in order to get a high-level recombination, especially from IQ, you need expression of both CYP1A2 and NAT2. We also have been looking at genotoxicity in terms of survival curves, and again, we've shown that if you look at a DNA repair mutant to get maximum sensitivity or toxicity of the compound, you need expression of both CYP1A2 and NAT2. And then we've also looked at other DNA repair genes involved in DNA damage tolerance, which have human homologs such as RAD18 and RAD5. And again, we see that expressing both CYP1A2 and NAT2 leads to maximum toxicity of the heterocyclic aromatic amine. Okay, so we've been looking, we wanted to extend these studies to look at other CYPs that could be colon-specific or expressed in the colon. We've been having some fortune with CYP3A4 that's expressed at low levels in the colon. And what you can see in this particular slide by this growth curve is that as you increase the concentration of IQ, you see more sensitivity when the strain is expressing CYP3A4. So I gave you a snapshot of some of the studies that we do, and just in conclusion, just want to mention that aflatoxin is a very strong genotoxin in yeast. There are biomarkers that include DNA adducts, RAD51 foci. We can use our yeast system for expressing cytochrome P450 polymorphisms, and so we can phenotype some of these polymorphisms in terms of how they activate these compounds. Heterocyclic aromatic amines, such as IQ, are recombinogens in yeast, and as I showed you, DNA repair mutants are IQ sensitive. And so we have developed these yeast libraries. We're developing some additional yeast libraries expressing other CYPs, but these yeast libraries are useful in identifying aflatoxin-sensitive genes and for screening other CYP-activated xenobiotics and perhaps other drugs. And the future directions, eventually we want to bring this back to mammalian systems, and we want to know whether the autologous genes, the mammalian genes, also confer resistance to these carcinogens. So just to acknowledge, I want to acknowledge a postdoc fellow in the lab, Min-Sing Sun, who did much of the work, and a medical student from Albany Medical School. I need to acknowledge Monica Keller-Seitz and Christian Sinkstad from the Swiss Institute of Technology, who helped me getting started with aflatoxin. Chris Volpe has been very important in terms of helping with the genomic studies and profiling and NIH funding. So thank you.
Thank you for your presentation. Is there any question?
Any questions?
Some Oslock gene confer resistance to AFP1, as you said. So what is the mechanism? Those genes metabolize AFP1 to inactive metabolites.
So
what is the mechanism?
Yeah, what's the mechanism? So for the
those genes confer resistance to.
Yeah, so for the DNA repair genes, it's fairly obvious that they're involved in repairing some type of DNA damage that is either involved in the direct damage of aflatoxin or indirect damage caused by aflatoxin. With other genes, it's not always clear what the mechanism may be. I'm not quite clear what the iron transport genes are actually doing in conferring aflatoxin resistance. They could be involved in sequestering the iron from the cytochrome P450, that's one gas, and making it slightly less active. And I think for all these different types of gene ontology groups, there may be slightly different mechanisms that could do either with the P450 activity or could deal with the transport of the aflatoxin into the cell.
Okay, thank you.
Any other questions for Dr. Fassulo?
So I probably missed it, but you said that aflatoxin is a genotoxin when it's activated?
Yes.
Okay. But then when you did the transcriptional profile with aflatoxin, that aflatoxin was not
No,
activated.
I'm sorry, that aflatoxin was activated. So,
It was activated.
So the strain actually did contain CYP1A2
Okay.
upon activation. And I should make a point that at fairly high concentrations of aflatoxin, even without the CYP1A2, you see some level of toxicity. So you see a lot more toxicity if you're expressing the P450, but without the P450, once you get above 100 micromolar levels of exposure, you do see some levels of sensitivity. So some of this aflatoxin is getting into the yeast cell, perhaps is intercalating in membranes or interfering with some function without being genotoxic per se.
Okay, thank you.
Any other questions?
Thank you very much for your presentation.
We're going to wait till after the session's over, and then we'll give everyone their
Okay.
certificate.
Okay, good.
Okay, so let's move to the second one from Silma Regina Ferreira from the Federal University of Maranhão. Is it correct? Maranhão? Okay.
Can you help here?
I am not familiar at all with this.
He's coming.
Please, where are those? Oh, sorry.
It's the fourth one. You operate? Okay. Okay.
Very nice.
Yes, thank you. Well, good morning, and I'm Dr. Silma Pereira from Brazil, and it's a pleasure to be here and have the opportunity to talk about the effects of glucantime on DNA. Glucantime is a pentavalent antimony used to treat leishmaniasis, which is an endemic disease in Brazil, especially in my state, Maranhão, located in the north of the country. This disease affects the most deprived social groups. For example, in 2013, 558 cases and 49 deaths were documented just in the state of Maranhão. Leishmaniasis is a neglected tropical disease caused by over 20 species of the protozoa Leishmania, which are transmitted by infected female phlebotomine sandflies. Wild and domesticated animals and humans themselves can act as reservoirs of infection. There are three main forms of the disease. The first one, visceral leishmaniasis, also known as Kala-Azar, is fatal if left untreated. It is characterized by irregular fever, weight loss, enlargement of spleen and the liver, and anemia. Cutaneous leishmaniasis is the most common form of the disease, and it causes skin lesions. Mucocutaneous leishmaniasis leads to partial or total destruction of mucous membranes, and the majority of cases occurs in Bolivia, Brazil, and Peru. Visceral leishmaniasis is widely distributed around the world and is a public health problem in 88 countries, and occurs especially in Brazil, India, Bangladesh, Ethiopia, Sudan, and South Sudan. And cutaneous leishmaniasis is more widely distributed. And about 1/3 of cases occurs in the Americas, Mediterranean basins, and Western Asia. Regarding treatment, pentavalent antimony is the first choice drug recommended by the World Health Organization to treat all types of leishmaniasis. However, there are many adverse effects observed, such as nausea, vomit, diarrhea. And these patients also may develop cardiac, liver, and kidney complications, especially at the end of the treatment. So, because of this, and knowing that the mechanisms of action and toxicity of pentavalent antimony is still unclear, we decide to investigate if and how this drug interacts with mammalian DNA. Trying to understand that, we performed COMET-INSEI in vitro using human lymphocytes and in vivo using mice leukocytes. Furthermore, regarding COMET-INSEI, we performed COMET-INSEI in vivo followed by FPG digestion, which removes oxidized DNA base. And to investigate mutagenicity, we carried out micronucleus tests using mice erythrocytes. In addition, we investigated if this drug could cause cell death, so we performed apoptosis assay. Previously, using systems with and without bioconversion, we demonstrate that the anti-leishmanian caused DNA damage in vivo, but not in vitro. Here in this table, we present the DNA damage score detected in human lymphocytes treated with different concentrations of glucantime for 3 and 24 hours. And as we can see here, we identified a difference for all groups treated with glucantime compared to the negative control. Cells were also exposed to the drug for 48 hours, allowing them to go through DNA repair mechanisms. So we could investigate if this drug could cause cell death by apoptosis or necrosis. Regarding apoptosis, we didn't find any difference for any groups treated with glucantime, but the drug was able to increase cell death by necrosis. So these assays, these results support those one obtained from in vitro COMET-INSEI. So glucantime wasn't able to cause DNA damage in vitro and apoptosis in vitro without bioconversion. On the other hand, in vivo tests using leukocytes from Swiss mice treated with glucantime for 24 hours and in macrophages for 3 hours showed an increase of DNA damage for all groups treated with glucantime compared to the negative control. And it's important to mention that we didn't find any difference between groups treated with glucantime and the positive control, which was treated with cyclophosphamide, a recognized genotoxic and mutagenic drug. So, knowing that glucantime was able to cause DNA damage in vivo, the next step was to investigate if the drug could cause mutations. So, we performed micronucleus tests. And here we can observe that glucantime was able to increase the frequency of micronucleated cells compared to the negative control. So, from these results, we suggest that glucantime is a promutagen that requires metabolization to exert its mutagenic action. Our data support the idea that pentavalent antimony is a prodrug that is transformed into the more toxic trivalent form, SB3, to exert its anti-leishmanian activity. Glucantime action was also evaluated using BALB/c mice infected by Leishmania infanto-Chagas, which cause visceral leishmaniasis. And we can see here that the infection itself increased DNA damage and the frequency of micronucleated cells. And when these infected animals were treated with glucantime, the genomic lesions and mutations further increased. In addition, the activity of superoxide dismutase was higher in the infected animals compared to the negative control. And we didn't find any difference between infected animals and those infected animals treated with glucantime, so suggesting the occurrence of oxidation in both groups, infected animals and those one treated with glucantime, which presented genotoxic and mutagenic effects. So, trying to investigate if glucantime itself could cause oxidative damage, we performed assays using uninfected animals. In this case, we performed Cometa assay followed by FPG digestions, again, which removes oxidized guanine. And here we can observe that DNA damage obtained using Cometa assay followed by FPG was higher than that obtained by conventional Cometa assay. And similar results were observed regarding the positive control treated with cyclophosphamide. So we suggest that glucantime itself does cause oxidative damage in DNA base. So to reinforce the idea that this drug could cause DNA damage and mutation by oxidation, we carried out antigenotoxic and antimutagenic assays using compounds that present protective effects against oxidative stress, such as isoflavones and vitamin C. Here we have some results from antigenotoxic and antimutagenic assays using Genstein, which is a type of isoflavone. And we can observe that in using Genstein was able to reduce the DNA damage, the frequency of micronucleated cells caused by glucantime, achieving similar scores and similar frequency of micronucleated cells observed in the vehicle control. And this reduction was up to 68%. Similar results were obtained when we treated these animals with vitamin C. We can observe here that vitamin C reduced the genotoxic and the mutagenic effect caused by glucantime. In this case, the protective effect was observed when both drugs, vitamin C and glucantime, were administered simultaneously or with post-treatment. However, when these animals received vitamin C prior to glucantime, the protective effect wasn't observed. And in this case, the reduction was up to more than 100%. So, in summary, we can say that glucantime does not induce DNA damage and apoptosis in vitro without metabolization. Infection by Leishmania itself causes mutation in mammalian cells. Glucantime increases even more the frequency of mutations in these infected animals, ACOD activity increase in infected animals treated with glucantime. So glucantime is a promutagen which requires metabolization to exert its mutagenic action. Glucantime induces oxidative damage in DNA base. And finally, genistein and vitamin C present high efficiency to prevent genomic lesions and mutations caused by the anti-leishmanial glucantime. So in conclusion, to answer our first question, we suggest that the anti-leishmanial glucantime does cause DNA damage by oxidative stress. But other questions arise, and now our group is conducting some experiments using bulb C infected by Leishmania infantis chagas. And these animals are being treated with vitamin C and glucantime for 20 days to determine the parasite load and to investigate the expression of some genes involved in the process, such as apoptosis, DNA repair, and oxidative stress. We are trying to investigate if vitamin C could be used as a co-treatment to protect the DNA damage caused by glucantime without reduce its anti-leishmanial activity. So I would like to say thank you for my group in Brazil, my students, and for the foundations in Brazil that have been support this research. And thank you for your attention. And now I'd like to open the floor to questions. But before we go, I want to show you some pictures of my city, my state, Brazil. And as you can see, we have more than leishmaniasis there. Thank you.
I'd like to ask a minor question. I was very surprised in your results when you presented the oxidative damage observed by the comet assay after
FPG digestion.
Right. Yes, right. What about the net oxidative damage? Because it is well known.
Sorry?
What about the net or the damage that is observed when you subtract the damage observed with your test compound after the treatment or after the exposure with the restriction enzymes minus the damage that the buffer of the restriction enzymes induced by itself? Because it is also well known that the buffer of the precisely FPG induces the highest rate of DNA damage.
Yes, but that's because of that we have compared our results from comet-assay using FPG with those one used by just conventional comet-assay that have the same compounds in the reagents.
Yeah,
and we have compared as well with the negative control.
Yeah, but you should also have included one treatment in which you have been only treated the cells with the buffer.
Yes, but
this is impossible to get such a
but
conclusion
we
then.
add the enzymes.
Yes.
In the end of the experiment.
Yes,
so.
I know. But in order to be sure that the enzyme recognized in this case the oxidized pyrimidines.
Yes, that's why we performed other experiments as with some antioxidant agents such as vitamin C and genistein and isoflavone, just to reinforce our first idea based in this experiment with comet-assay followed by FPG. So we can't say just with this assay that we have oxidative stress, but we have another results using compounds that have protective effect against oxidative stress that reinforce our suggestion.
Why didn't you include such experiments that is almost
Yes,
masked
Yes,
in this type of
we
research?
don't we didn't finish yet. Ne? We are performing another experiment, including infected animals treated now with vitamin C and glucontime for 20 days. And we are conducting some experiments, including activity of some enzymes involved in the oxidative stress. Okay?
Okay, thank you.
You're welcome.
Any other questions?
Just how effective is glucontime at treating the leishmaniasis infection?
Sorry.
How effective is it in the treatment of the disease?
We're on time.
Which cause leishmaniasis?
Yes. How effective is glucontime? If it's taken at the right time, is it effective in the treatment?
Actually, unfortunately, in many patients, they are diagnosed just when the infection is kind of high, so the prognosis is not very good. But for a human treatment, glucontime is administered for around 20 to 30 days. So the patients, they shows many adverse effects. And we have some related case of many children that presented leukemia after the treatment. So that's why we start to investigate if this drug could cause some mutations on DNA.
Thank you, Dr.
Yeah.
Thank you.
So let us now call Dr. Arpan Elnar from the University of France.
To come up and load your talk, so. Did you get it?
Yeah.
Great.
So a little bit of background about PCBs. Among persistent organic pollutants, around 1.5 million
Where is the mouse? Sorry. Let's try it again. Which one is it?
It's Toxicology Summit. No, the second one.
That's why. So it was not our problem. Do we need another computer? I have one. Why are you not working? My own computer. No. This is not for
Do you have your thumb drive?
Switch to another computer? No, we cannot. I can use mine as well. Video. We should be sure that he's not here.
That's what I said.
Is Dr. Tilak here? Yes.
Is he here? Dr. Tilak? Does anyone know him?
From India.
From India, Dr. Tilak?
So if Dr. Tilak is not present, okay, Dr. Ulujimi will present his data instead of him. What doesn't mean he will move from the afternoon till very early in the afternoon, at 12:10. Yes, 12:10.
He's not here, so I'll make sure
Ah,
I'll find
he is not here as well.
No, I mean, I'll find him. He's not sitting in the room. I'll find him and tell him
Okay.
so that he's prepared for you.
Thank you for your cooperation.
Hey, we do what we can do.
Yeah. Yes.
Let's do this.
Uh,
Since
why
we're having
don't-
technical difficulties, let's take our short break now. Just take 10 minutes, and then when we come back, and
Yeah.
I will come out and get you if you don't come back here. So in 10 minutes, just so go, have something to drink, use the facilities, and come back so that we can.
I
get
couldn't
on
do
the
it in, I
presentation.
couldn't say in this way, but,
So enjoy your break, and I will look for you back in 10 minutes, okay?
including me.
You first. Me first. You first, because we have to get you up here.
Okay.
We're so sorry.
Yeah.
So sorry. Oh, I don't mind. I asked him. Oh, you already did.
Okay.
All these brilliant people. All right, I'm gonna go look for all of them. Yes, please. You mean you and then. Okay, exchange, yes. You can write, or, no, see your email is here. And I think I have mine too, so can you send me my presentation? My mail is here. Okay. Okay, can you send? Yeah. Okay, so,
It's just, it's not loading hers for some reason. It's on there, but it doesn't go full screen. This doesn't work on your laptop, so, you see my problem?
So among persistent organic pollutants, over 1.5 million tons of PCBs were produced worldwide under international names such as Aerochlor. They were produced mainly for their industrial use and technical applications, such as electrical transformers and capacitors. But despite being banned since 1970s, they are still leaked into the environment from old electrical equipment. They comprise 209 congeners, distributed into subtypes according to their conformation. We have 12 coplanar called dioxin-like PCBs, which are highly toxic and very well studied, and 197 non-coplanar congeners called non-dioxin-like PCBs that are predominantly present in the environment due to their occurrence in environmental tissues and animal tissues. So PCBs represent the highly intake among pups, reaching 615 nanogram per day. And PCB contamination is mainly due to fish consumption. As you can see here, fish contains 9% of dioxin-like PCBs and 91% of non-dioxin-like PCBs, in which the sum of the 6 non-dioxin-like PCBs represent 56%. So in this 56%, we noticed that the PCB 153 and 138 are the most predominant in fish matrices, reaching 37% and 32% respectively. So we are mainly exposed to PCBs through ingestion. And once absorbed into the organism, these compounds passes through gastrointestinal tract, and they are metabolized, undergoing metabolism phases 1 and 2, and especially stored in fatty tissues, including brain. They are eliminated by feces and urine, but mainly also transferred by lactation to the children or newborn babies. So breastfeeding constitutes a source of elimination of PCBs from the mother to the child. Studies have shown that 95% of PCB 153 are transferred to the newborn babies during lactation, and only 5% are transferred to the fetus. So it could be also a source of elimination because, for example, PCB 101 body burden could drop in the mother around 75% for the third child during lactation. So breastfed babies might receive high levels of PCBs during a critical phase of their development, and where the central nervous system is particularly vulnerable to these xenobiotics, and that juvenile brain is immature compared to the adult brain. Many epidemiological studies have correlated the concentrations detected in the maternal blood and milk to the neurological impairments or deficits observed in the children. So mainly learning and memory deficits and motor development impairments. Studies have shown also that dioxin-like PCBs could bind to the aryl hydrocarbon receptor and cause neurotoxic response. But non-dioxin-like PCBs do not bind to this receptor and could have the same mechanism of action like dioxin-like PCBs. But this is only a review of Codovanti, who supposed that non-dioxin-like PCBs could act also as the same mechanism of dioxin-like PCBs and induce the same effect. So many tolerable daily intakes were fixed 10 years ago, especially on dioxin-like PCBs, because they have the same mechanism of action such as dioxin and could have neurotoxic effects. So, and many experimental studies have shown that there's these neurotoxic responses. So TDI of 20 nanogram per kilogram per day was established for the dioxin-like PCBs. And we calculated the dose for the non-dioxin-like PCBs, since the non-dioxin-like PCBs constitute 50% also of the occurrence of these congeners in the total PCBs. So it's only a calculation. It was divided by 2 to get 10 nanogram per kilogram per day for the non-dioxin-like PCBs, but there were no experimental studies on this dose. It's only a calculation. So our question was, what are the short and long-term effects of lactational exposure to this mixture on juvenile neurodevelopment? We wanted to evaluate the neurotoxicity of this mixture at low levels especially, focusing on mechanisms underlying the developmental neurotoxicity of this mixture, using a murine model and characterizing mixtures and not only single compounds, which reflects the environmental exposure. So our protocol started with exposing dam mice to this mixture, starting parturition until weaning of the pups, 21 days, and by gavage to this mixture according to the constitution or the distribution of each congener in the mixture found in literature in fish matrices. And we were fixed on TDI fixed in a human, which is 10 nanogram per kilogram per day. And we have fixed 3 levels below this TDI to see if there is some neurotoxic responses. So this is the general scheme of our study. Today I will focus only on the transcriptomic analysis or RNA expression of genes in the brain. And at PND 14, because it's the end of exclusive breastfeeding of the dams. So to do that, we have isolated neurons by magnetic separation from cerebellum at PND 14. We had done the RNA labeling, check the integrity of RNA, and analyze with GenSpring the transcriptional changes. So we have chosen also the cerebellum because it is implicated in ocular motricity, locomotion, and cognition. And we focused only on one dose because this dose had the most significant effects observed in male mice. And that's why I will show you some of the results in the functional level, at the functional level. So the main results was at PND 14, when we observed that male and female mice passes more time to go to the open arms compared to the control in the test of elevated plasmase, which evaluate the anxiety of the mice. This effect was persistent because on PND 160, we conducted another test, which is the light-dark box test, which have the same principle than of elevated plasmase test. And we have seen the same effect on an advanced life stage of the mice. We have also seen an overexpression of ryanodine receptor, which is a rhinoceptor involved, implicated in the non-dioxin-like PCBs toxicity in literature, and especially at 10 nanogram per kilogram. So we have published these data, and we have concluded that there are some transient disturbances, longer turning reflexes in male pups, reduction in their general activity, but the most important result was the persistent anxious behavior observed at more advanced life stage. Now, let's look at the results concerning the transcriptomic analysis. We have shown that, we have observed that 693 genes belong to terms involved in cell cycle and response to DNA damage stimulus were overexpressed. And downregulated genes belonged to genes involved in neuron projection and neurotransmission were also found compared to the controls. So we wanted to validate our transcriptomic analysis by using qPCR technique and with selecting genes involved in DNA damage. And we have observed that these genes were overexpressed also in the mice exposed to 10 nanogram per kilogram, as you can see here. At the protein level, we have also found that there were some proteins which are implicated in the transmission of electrical signals of neurons were down or upregulated. So these findings could supply or our analysis, our previous analysis or results. In summary, we conducted gene expression profile of cerebellum in mice exposed to the mixtures at low levels, and we have found that it induces transcriptional changes in neurons. And some of the genes were downregulated, belongs to response to DNA damage and cellular stress. Some of the proteins involved in the conduction of electrical signals were also decreased. So general discussion was, is the non-dioxin-like PCBs could act like commercial mixtures, such as Arachlor 1254? Maybe it could be, because it could underlie mechanisms of genomic changes and functional changes, as we already published. It arises also other questions. Is TDI of 10 nanogram per kilogram of NDL PCBs is really tolerable? Maybe we need more studies to prove that. Is eating fish safe? A lot safer than not eating fish? We have really focused on this thematic since 2 PhD theses were started 2 years ago. One of them was the multi-generational epigenetic study of the exposure to these NDL-PCB mixtures. Another one is the effects on neuroinflammation. We had also evaluated the effects of fish consumption, especially eel matrices, on the central nervous system development. This project was accepted by ANR, but the data were not published. We have also think about the preventive approach, using antioxidant-rich food to deal with the deleterious effects of contaminants, for example, quercetin. I would like to acknowledge my team in MES, and especially my professor, and also the financial support from ANR and also FEDER. And really, thank you for your attention.
I just want to ask you that question back. Do you think it's safer not to eat fish?
It depends on the consumption of the eel, of the fish. For example, the type of the fish. So, for example, eel matrices are have bioaccumulative properties, especially for the non-dioxin-like PCBs. But we can, every type of fish has his own profile. So it's difficult to respond to this question.
It's very difficult for the public to think about its profile before they start to eat it.
Yeah.
So, I mean, it's an issue, really, isn't it?
Yeah.
I want to ask about the administration. You give a dose every day for 21 days.
Yes.
And you use 2 models. plasmase and dark and light.
Yeah.
And it looks like that dark and light is more sensitive. Didn't you notice that?
Box is a more sensitive test.
More
yes.
sensitive because the lower dose produce significant effect.
Yes, that's right.
Okay, thank you.
But there are so many variables. So maybe only one variable could has been shown also in the in the elevated plasmase test we have for the 3 doses, not only for 2.
I think we have time for 1 or 2 more. One more.
Just a quick comment and a question. So the comment is that the Hudson River, which I don't live too far from, in New York, is actually heavily contaminated with PCBs many years ago. And so now they're doing studies, and they do allow you to fish from the Hudson River, and there's a certain quota. So it may be interesting in further studies to look at the epidemiological studies that are currently being done in terms of fish consumption from the Hudson. The question I had is that you mentioned that these compounds were actually activated. And I wanted to know if there had been in vitro studies in which you just add the compounds directly to cell culture, and do you see upregulation of these genes?
Not yet, because our main topic is environmental exposure. It's not really in vitro exposure. There are so many studies that are done in vitro, but not in vivo like we did.
Thank you very much, Dr. Elmer.
Action of L-ascorbic acid and green tea infusions in the acute toxicity and mutagenicity of reaction mixture nitrate-sulfonamide. Nitrite is a normal component of the organisms, and it is in cured meat as preservative to in contaminate drinking water. Nitrite can react with nitrogen compounds in acidic media to give N-nitroso compounds, potentially mutagenic and carcinogenic. N-nitroso compounds can be exogenous or endogenous. Since the pioneer studies of Lisinski et al., numerous papers dealing with the chemical and biological aspect of drug-nitrate interaction has been published. However, the problem of endogenous drug nitrosation is largely unrecognized. Sulfonamide and their derivatives are widely used for their various properties, antibacterial, antifungal, both in human medicine and in veterinary, antidiabetics agents, diuretic, herbicides, analytical reagents, etc. So it is common to find them as contaminants either in the environment or in foods of animal and or plants. In addition, there are among the antibacterial agents most widely used in the world, mainly because of its low cost, low toxicity, and good antimicrobial activity. We can see some sulfa drugs in clinical use today, phthalate sulfatiazole, sulfanilamide, sulfamethoxazole, glibenclamide, gliburid is antidiabetic. Because sulfonamide possess amines and amide functions in their molecule, they can react with nitrate, giving N-nitroso compounds, potentially mutagenic and or carcinogenic. This is a primary amide, secondary amide in sulfatiazole and in glibenclamide. Natural compounds having anti-cancer or anti-mutagenic activity are chemopreventive substances. L-ascorbic acid and green tea inhibit nitrosation reaction by reaction with nitrate. This lecture is taken from part of the PhD work of Dr. Ana Pontoriero entitled Chemopreventive Action of L-ascorbic acid and green tea extract on mutagenicity and acute toxicity of reaction mixture sulfonamide nitrate for the following sulfonamide or their derivatives: sulfatiazole complex, sulfatiazole cobalt 3, and gliburid. This talk describes the work with sulfatiazole nitrate mixtures by the Ames test and glibenclamide nitrate mixture by the Ames and allium test. For both mixture, we will analyze UV spectra. The work conditions in this work is pH 2, room temperature, molar radio, nitrate, sulfonamide 3 to 1, but in 112 is react. The concentration of anti-mutagens from 0 to the concentrations of nitrites, and addition of anti-mutagen was before and after the nitrite. And the biological studies was the Ames test and allium cepa test, and physical chemical studies, UV spectra. Well, the Ames test we saw recently. This is a photo of spontaneous revertance on TI 100 strand and revertance after treatment with the nitrate mixture sulfatiazole nitrate in acidic medium, and the same previous photo showing the manual counting of revertance colonies. The effect of the antimutagens, L-ascorbic acid and green tea extracts on the Ames test. Well, we saw ascorbic acid did not mutagenicity in the Ames test with both strains. And green tea extracts did not show mutagenicity in the tested conditions in the Ames test with Salmonella typhimurium, TI nitrate strain and Tia102. This is the formula to calculate the percentage of inhibition of mutagenicity, and we can see we will see the mixture sulfatiazole nitrite. The ascorbic acid is proposed that reacts with nitrite reduce the nitrite to nitric oxide. The reaction mixture sulfatiazole nitrite show mutagenicity in the M stage with the Tia98 strain and with the Salmonella typhimurium Tia100 strain 2. Well, ascorbic acid added before nitrate inhibit mutagenicity. We can see with the Tia98 strain. And if ascorbic acid is added after the nitrite, inhibit mutagenicity too with the same strain. And with the strain Tia100, ascorbic acid added before nitrate inhibits mutagenicity, and ascorbic acid added after nitrate inhibits mutagenicity too, with the two strains. We try with green tea. Green tea is an inhibitor of nitrosation reaction by reaction with nitrate. The aqueous green tea extract treated with nitrate in acidic medium show no mutagenicity in the Ames test. Green tea is rich in catechins, epigallocatechina, galat-catechin present in greater proportions in green tea. The effect of green tea in the sulfatiazole nitrite reaction mixture. Green tea added before nitrate inhibit mutagenicity with the strains Tia98, and the green tea added after nitrate inhibit mutagenicity too with the same strain. As a green tea aqueous extract without dilution added to the reaction mixture before or after nitrate inhibit the mutagenicity activity of the mixture. From the first added, we decide to try it diluted 1 to 10. And green tea diluted 1 to 10 added before nitrate inhibit mutagenicity with the strain 98, and green tea diluted 1 to 3 added after nitrate inhibit mutagenicity with the same strain. We tried with glibenclamide. Gliburide is anti-diabetic. Increased concentrations of glibenclamide did not influence over the coefficients of reversions of Salmonella typhimurium strains Tia98 and Tia100. Glibenclamide did not show mutagenicity in the Ames test in the tested condition. Reversion coefficient is like 1. The mixture glibenclamide nitrate reaction mixture did not show mutagenicity in the Ames test with Salmonella typhimurium 98 strain in the tested condition and in Tia100 strain 2. We tried the mixture glibenclamide nitrate reaction mixture prepared according to the conditions suggested by the WHO for the study of a drug nitrosability did not show mutagenicity in the aim stage with Salmonella typhimurium TIA 98 and TIA 100 strain. So, as a glyburide nitrite mixture in acidic media did not show mutagenicity behavior in the aim stage, we decided to try in the allium cepa test. The allium cepa test is a biological assay using bulbs of allium cepa L to determine phyto, cyto, and genotoxicity of substances in liquid media, waters, effluents, several chemicals, medical drugs, etc. The plant's genotoxicity test, the allium cepa test, is commonly used widely in laboratory texts because its rapid growth of its roots, the response of its genetic material to the presence of potentially cytotoxic and genotoxic substances in liquid media, and a high correlation with mammals test system. Another advantage of this test system is the presence of an oxidized enzyme systems that is essential for pro-mutations evaluation, besides its low cost, simplicity and non-sterile conditions. So, equal size young balls of common allium cepa were used. Onion balls, 7 per dose, were kept in mineral water for 48 hours, then exposed to glibenclamida and glibenclamida nitrite mixture. Conditions at root temperature and protected from the light. The parameters evaluated in the allium test was macroscopic parameters, room elongation, and presence of abnormalities as blue hoop, necrosis, tumors, and microscopic parameters, mitotic index, chromosomal aberration, nucleus abnormalities, and micronucleus. The length of the root as an index of toxicity and modifications in root consistencies and shape, formations of two more hooked root, twice root were observed are macroscopic parameters. This is the zone for measuring the mitotic index. The mitotic index was determined by scoring more than 5 millicents per slide. The mitotic index was calculated as the number of dividend cells taking into account the following mitotic phase, meta, ana, and telophase for 1,000 observed cells. Mitotic index, the decrease or increase or decrease, is an indicator of cytotoxicity. The chromosomal aberrations indicate genotoxicity and change in the number of chromosome structure. This is a picture of our work where we can see a chromosomal aberrations in allium cepa in meristematic agent exposed to chemical agent. This is normal anaphase and anaphase with chromosome breach in the sulfadiazole nitrite mixture. The staining is a Schiff reaction and the zoom. In the glibenclamide nitrite, glibenclamide did not show influence on the root length in allium cepa in the tested condition. The p-value is more than 0.05. And glibenclamide, yes, increased mitotic index of allium cepa in the tested conditions, a little, but increased. But glibenclamide nitrite mixture did not show influence in the root length allium cepa in the tested conditions. Glibenclamide nitrite mixture did not show, it's the same. Comparing mitotic index shows decrease in the mixture. An increasing amount of ascorbic acid with a constant dose of glibenclamide nitrite mixture did not show influence on the mitotic index of allium cepa, the p-value more than 0.65, then we may suggest that there was not production of cytotoxic substances with the addition of ascorbic acid. The spectroscopic analysis of sulfa nitrite mixture and the antimutation ascorbic acid and green tea. In the ascorbic acid solution was equimolar with nitrite, the sulfadiazole nitrite mixture. The spectral patterns of sulfadiazole nitrite mixture plus ascorbic acid with the same both ascorbic acid being added before or after the nitrate is if when ascorbic acid increase the concentration, the absorbance in 357 nanometers decrease. And with green tea and the same mixture, this is the spectra obtaining measurement, total polyphenols in a green tea infusion in the presence and the absence of reaction mixture sulfadiazole nitrite, this is green tea, and the mixture green tea with polyphenols diminished. The spectral pattern of sulfadiazole nitrite mixture plus green tea was the same, both green tea being added before or after the nitrate. Here we can see increase with green tea, the absorbance at 417 nanometers increased too. So we did differential spectra. In the differential spectra of the reaction mixture with increasing amount on green tea minus the sum of the spectra of the mixture and green tea alone, show increases of absorbance at 417 nanometers and decreasing at 357 nanometers. In the electronic spectra of glyburide in the presence and the absence of nitrite did not show evidence of reaction. Summarizing, the spectra analysis allows us to suggest the following. The reaction between sulfadiazole and nitrite occur instantaneously in acidic media. The antimutagens, ascorbic acid or aqueous green tea extracts, reacts with the formed mutation instantaneously, and its action is independent of that it is added before or after the nitrate. Glibenclamide, commonly used antidiabetic, does not react with nitrite in the tested condition, confirming the not observed mutagenicity in such mixture by the Ames test. So, as conclusion for everything mentioned above, both the ascorbic acid and green tea appear to be efficient antimutagens in mitigating mutagenicity of sulfa-nitrite mixture in acidic medium. While this study is only an initial stage, it is an auspicious to reach suggests that oral administration of a sulfonamide is conducted jointly with ascorbic acid, vitamin C, which may or may not be added to the excipients, or with a cup of green tea to reduce the risk of sulfur reaction with nitrates in the acidic medium of the stomach. I show where is Rosario in South America, Argentina, and another view of the Rosario and some people from Rosario for the football games is Lionel Messi, yes, Luciana Aymar for hockey and the river coast. And this is my group, the sulfas group. This is Anna, we in the faculty, and Mary with the onions. Well, thank you very much.
Dr. Risotto, do you think it's valid, your extrapolation from AIM test to humans, and to suggest such conclusion?
Yes, we saw that the reaction mixture was sulfa with the sulfatiazole and nitrate was a very, very mutagenicity. It is formed a direct mutation because we did the AIMS test without metabolic activation, and we analyzed too in the allium test. So we think that it's very it's a high risk of nitrosation or reaction. I don't know if it's a,
yeah, I mean, but
a product of nitrosation. It's a mutation.
Okay, but regardless of the mutagenic effect you were able to detect, no synergisms nor either additive effect, do you think it's valid to directly say that you could suggest to blah, 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 and not going through different steps that I think
no, yes, it is a first study, so, but we think that is the two anti-mutations have a good effect in mitigate the action of the possible mutation produced in the mixture, and are so good to, but the risk of a reaction of nitrosase of drugs that can be nitrosated is a high risk.
from Shandong University. The title for this presentation is exploring mechanism of oxidative damage and cell apoptosis induced by lead explorer. Yeah, a little difference from the conference book. I will go the presentation within this book. Okay, with this 4 parts. As you know, lead can exist in dust, water, soil, food, sediment. It can come inside to our body. Yeah. As reported by World Health Report, lead is the top 20 risk factors to our health. That's why we are focused about the lead. At different dose, it can induce different risk. At a low dose, it can induce headaches, weight loss, reproductive problems. At high dose, can decrease our memory and kidney problem, even cancer and death. So that's why a lot of people, a lot of researchers are focused about the risk of lead. For the first part, we just care about lead-induced oxidative stress. For this, mainly on the SOD is in the very important key part of oxidative stress. We did an experiment on the zebrafish level and in vivo and in vitro on the SOD. So how lead interact with OD during the oxidative stress is an enzyme but not a biomarker. Here's our experiment formula. Use the zebrafish to get the tissues of livers and then get the cells under the experiment. This is the experimental result. We can see lead can decrease the amount of glutathione and increase the amount of GSSG and the MDA. Is obviously the oxidative stress occurred after lead exposure. Okay, here's what we are concerned about for the first few questions. Does lead interact with SOD with similar way both in vivo and in vitro? How does lead influence the activity of SOD? The mechanism of lead decreased the activity of SOD. Okay, we already showed this for the after exposure of a lead to zebrafish, and we can detect the activity of SOD decreased. Even for the in vitro experiment, the exposure of lead directly to SOD, the activity still decreased. The direct interaction between lead acetate and the purified SOD caused the activity of SOD decreases significantly with after the addition of lead acetate, corresponding with SOD inhibition assay in vivo. Okay, like this one. The change of the activity may be induced by two factors: the structure change of SOD or the amount change. So the SDS PG result showed that the exposure of lead not induced insignificant changes in the expression of SOD. So for the direct interaction, we monitored by the fluorescence technique. The results showed that after their interaction, the intensity of fluorescence decreased, but there is no peak shift. This is the structure of SOD. Is the active site right at this side, mainly closely related to copper and zinc irons. To further probe their interaction mode, we applied the lifetime of fluorines. The fluorines lifetime decreased and showed that they are static quenching process after their, the direct interaction of lead with SOD. The ITC result still detect the direct interaction of lead with SOD. The result showed that the binding is mainly through the electrostatic binding. The delta G with negative delta G showed us that they are spontaneous interaction. For their structure, their structure change, the UV and the CD results showed the skeleton conformation and the secondary structure of SOD were changed after the binding of lead. As mentioned of slides before, we know that copper and the zinc irons located in the active site of the of SOD. After their interaction of lead with SOD, the activity decreased. We think we are not sure if lead will replace the metals of copper or zinc. The experimental result showed that after the exposure of lead, more copper and zinc irons freed to the solution. They leave their active site. Copper, you see, after 10 microgram and 50 microgram per liter of lead exposure, 12.2% and 15.3% of copper released from SOD, and still zinc released from the active site. Maybe that's the reason their activity decreased. Okay. The ITC and the molecular docking results also illustrated that lead bound to the active site channel of SOD. Just at the active site, it interacted with adenine 141 and released some of the zinc and the copper to decrease the activity of SOD. Yes, this experiment showed that lead can interact with SOD both in vivo and in vitro with the similar way. Lead interacted with adenine 141 of SOD through electrostatic forces, hindered the path of the catalytic substrate and released copper and zinc to decrease the activity of SOD. The second part is the lead interacts with DNA in vivo and in vitro during the oxidative stress. Okay. For this part, the single cell electrogel result showed that lead irons cause a dose-dependent increase in the frequency of DNA breaks of the in vivo result. For the in vivo result, we still showed another experimental result of DNA protein cross-link result. This is an indicator of DNA damage in severe level. The DPC coefficient reaches up to 12.6% after 100 microgram lead, micromolar, micromolar of lead exposure. lead could not enter into the liver cells, we detected by the other experiment, but it can freely induce the oxidative stress, then induce the oxidative damage of DNA within the cells. For the direct interaction of lead with DNA, we applied this probe, MethBlue, as a probe. For the 3 binding modes, intercalation, electrostatic, and group binding, we used the ITC experiment and molecular modeling. This is the spectral result, and showed lead can interact with DNA. The iron strength influenced the fluorescence intensity of lead. MBDNA and MBDNA showed their interaction is influenced by the charge. So the main interaction should be electrostatic binding. Okay. The binding models, the ITC result showed there are 4 binding sites of lead with DNA. For site 1 and site 3 is the electrostatic binding. For site 2 and site 4 is the minor group binding. So from this research, we can see lead could not enter into liver cells freely in normal conditions and could not cause DNA damage through direct interactions, but indirect interactions with ROS, reactive oxygen species, produced after lead exposure. The ROS will induce the DPC. Lead iron could bind to DNA with 4 binding sites for their direct interaction, mainly by the minor electrostatic forces and group binding effects. So after the exposure of lead to the organism or cells, they will induce oxidative stress and produce a lot of reactive oxygen species. So there are 2 signal pathways, ERK, which is closely related to the apoptosis activated by ROS production. And the campus-3 is closely related to the cell apoptosis. So we think how the lead exposure will induce ROS and cell apoptosis. So we did the experiment on these 2 pathways. So our question is, can lead activate campus-3 and the ERK signal pathways to induce kidney or liver apoptotic process to reduce cell viability? The result is for the hepatocytes and the neurocytes. So, nephrocytes. Okay. So for the hepatocytes is decreased, but for the nephrocytes is more obvious. It indicates the lead to kidney cells were more sensitive. Yeah. This result is done by the Muse cell analyzer. Yeah. For the ROS production in liver and kidney cells, in the liver cells, we can see the lead exposure induced obvious ROS production. And for the kidney cells, it's still increased, but not so obvious compared to liver cells. Yeah, this is the amount of DSH, just to show the oxidative procedure. For the apoptosis result, we can see lead can induce obvious apoptosis in the kidney cells, and still increase the apoptosis process, but not so obviously. During the lead exposure, the ERK signal pathway activated greatly in the liver cells, but increased not so obviously in the kidney cells. For the ROS generation, is correspondingly after the ERK signal activated, more and more ROS produced. And for the CAPS3 activation, we can see after lead exposure, the CAPS3 in liver cell increased but not obvious. For the kidney cells, the CAPS3 activated obviously. That's why it will induce more cell apoptosis of kidney cells, and the kidney cells is more obvious to the lead exposure. Lead exposure can cause excessive ROS production in liver cells and highly activated ERK signal pathway, but the activity of CAPS3 remained stable, which results in lower apoptotic status. ROS contents in kidney cells presented highly increased after lead exposure, which could not regulate the activation of ERK, but CAPS3 was highly activated. So lead induced more percentages of apoptotic kidney cell levels than that in liver cells. Okay. Yeah, maybe somebody may be interested for the assistant professor or professor position available in our school, School of Environmental Science, Shandong University. Yeah. I really think for the financial support from Natural Science Foundation of China, the Ministry of Education, EPB in Shandong province, and STC from Shandong province, and the support from Shandong University. I really think for my collaborators, my postdoc, and my students work with me. Thank you very much for your attention. Okay.
Thank you for your presentation. I would like to ask you, first in your presentation, you mentioned zebrafish.
Yeah.
Zebrafish and the level of lead exposure in its intestine, liver, and kidney. And in the results, you only mentioned
for the kidney is
yes.
for mouse, not zebrafish.
No, you said for zebrafish you investigated the intestine, liver, yeah. and kidney. And in the result, I don't see any results about the intestine. So what about Caspase activity in the intestine?
For the liver and the kidney, this
is you discussed very well for the liver and kidney. My question is about the intestine of zebrafish. Okay. Is there any change regarding Caspase activity in it? Any apoptotic change caused by lead in the intestine?
You mean for the zebrafish?
For the zebrafish, yes.
Yeah, for the zebrafish, we didn't do the experiment of apoptosis.
Yes.
The result for apoptosis is for the mouse.
Okay, and how you investigate the intestine of the zebrafish?
You mean this? Yeah, you mean this part?
No, before, yes.
Yeah, but the first part.
The first part, yes.
This one?
Yes.
You mean which part?
You examine first above to the left the intestine. You take the intestine, the gallbladder, the spleen, the liver.
Yeah. For this one.
Yes.
Digestion.
Yes.
Yes, we did digestion. For this, get the liver, then cut them to a small piece, then digestion, and the centrifuge to get the cells.
Okay.
Then do the experiment.
Is there any positive results?
He's just pointing out where the destination.
Pointing, yes.
He's only just doing the liver.
You only focus on the liver. You only focus on the liver. You don't do the intestine, gallbladder, and spleen. Okay. Thank you.
Excuse me. Okay. You mentioned that lead. Whenever you increase the dose of lead, there is increase in copper and zinc. And this will lead to decrease the activity of SOD. Yeah?
Yeah, we think, yeah.
I didn't get it because copper and zinc, it's very important for the structure of SOD.
Right.
Yes? So whenever copper and zinc increase, the activity may be increased. I didn't get it, I need more explanation.
After the pull of lead to SOD, we can detect the zinc released from SOD.
Where did you detect it?
Oh, yeah. We detect, then we filter it. After lead is pulled with SOD, then we filter and detect the
you mean the copper and zinc becomes free?
Right.
From the, okay, thank you.
Another question?
So a comment question. Okay, so the comment is, I'm curious whether lead, like other heavy metals, nickel, causes epigenetic effects, because nickel causes a lot of epigenetic effects, so I'm just curious about that. The question is, relating to SOD, is there's the mitochondrial SOD as well, that's manganese, and I'm curious whether you've looked at if lead can also displace other divalent cations like manganese from the mitochondrial SOD.
You mean the...
I mean, the SOD you looked at was the cytoplasmic SOD, the copper zinc. And I'm just curious whether you looked at the SOD associated with the mitochondria. You haven't done that yet?
Not yet.
Okay.
One more question.
Kind of following up on the two previous questions. Is the lead binding stronger than the copper and the zinc to the SOD? Is it a much stronger binding affinity?
Yeah, it's much stronger. We compare, if without the lead is poorer, we can't detect the copper and the zinc released from SOD.
Okay.
Yeah, we did the experiment.
Okay. Okay.
Thank you.
Okay, in this case, we move to the next paper presented by Dr.
Okay, just as the time, maybe send this award to Fiona. Okay, yeah, I can just use this minute to send this award to Fiona. Yeah, for you. Yeah. Okay. Yeah. Congratulations for a good presentation.
were two objectives. First one was the assessment of the immunotoxicity for subchronic exposure to acephate, and then it was the assessment of the immunomodulatory effect of Acranthus aspera and Phyllanthus neruri in white leghorn cockerels. The study was conducted on day-old healthy male white leghorn chicks. Total births were 220, and the births were maintained as per the Institute Animal Ethics and CPSA guidelines, and all the necessary managemental procedures were adopted to keep the birds free from stress. All the chicks were vaccinated against the Ranichet disease with RDF strain at day 1 and 30 to check the antibody titer against NDV in serum. Similarly, the birds were also vaccinated against the IVD using gumno disease vaccine at day 14. And these experimental birds were vaccinated with Marek disease bivalent vaccine at day 1. Now, the total birds were divided into 11 groups, containing 20 birds in each group, and these were C1, C2, T1, T2, T3, T1 plus AA, T2 plus AA, T3 plus AA, T1 plus PN, T2 plus PN, and T3 plus PN. Now, the birds in the control group received only standard feed, and they have been considered as plain control, while the birds in the group C2 received groundnut oil and considered vehicle control. And the birds in the T1, T2, T3 received acephate in different dose levels, and the dose levels were divided on the basis of their LD50, and these were LD50 by 40, LD50 by 30, and LD50 by 20. Similarly, in case of the plant, the plant, the birds were exposed with the same acephate and concurrent exposure of the Acranthus aspera, that is for 1 milliliter per each group, and the same in the birds from the group T1 plus PA, T2 plus PN, and T3 plus PN were concurrently exposed with acephate as well as the Phyllanthus neruri, means alcoholic extract of the Phyllanthus neruri. Now, the blood was collected at 20-day intervals from day 0 to at the end of the study, and at day 50, the primary sensitization was done by DNCB, and it was challenged after 7 days for secondary sensitization, and at the last, these birds were sacrificed by dislocating the occipito-atlanto joint. Now, the collection of blood, the blood was collected in the heparinized as well as the non-heparinized tubes for hematoimmunological study. And at day 0, this blood was collected by directly puncturing the heart, and then the blood was collected from the jugular vein up to day 40, and then this blood was collected from the wing vein up to the termination of the experiment. Now, and in the last, these animals were sacrificed at the end of the experiment. The parameters which were evaluated were live body weight of experimental birds, immunotoxicological parameters, and these included humoral immunity, cell-mediated immunity, splenocyte proliferation assay, and organ body weight ratio of lymphoid organs, and in the last, histopathology of lymphoid organs were conducted. Now, the obtained results were analyzed by using the structure analysis, and for evaluation of the antibody titers against NDV, log transformation of the values was done. The statistical analysis of variance was done for factorial design, and one-way analysis of variance was used to compare the effects of treatment of various dosage of acephate on different immunological variables in the control and treated birds. The results were, this table depicts the effect of daily oral administration of the acephate on live body weight in white leghorn cockerels. There were no significant difference in the at day 0. This depicts that the experimental birds were of similar, approximately same weight, and they have been divided into different groups randomly. While the live body weight at day 7 was significantly reduced in the T2 and T3. These are the groups which received maximum, medium, and maximum concentration of acephate. And the live body weight was subsequently significantly reduced from day 7 to up to end of the study. And these reduction in the live body weight was effectively ameliorated in the birds which were concurrently exposed with the acephate as well as the alcoholic extract of the Acantha's aspera and Phyllanthus neruri. And these are similar pictorial presentation of the same. Now, this table depicting the effect of the oral administration of acifate on antibody titers against the RDF in white leghorn chicks. So there is no difference at day 0, while it was significantly reduced in T2 and T3 group from day 0 to day 60, while there was non-significant increase in the T1 group. And this reduction in the antibody titer against the RDF strain was effectively ameliorated in the group which was concurrently exposed for the acifate as well as the alcoholic extract of the Phyllanthus neruri and Acantha's aspera. This is the same. Now, this table is depicting the effect of daily oral administration of acifate on increase in skin thickness upon sensitization by the DNCB dye in white leghorn cockerels. So there is no significant difference in the treatment as well in comparison to controls after 6 hours, while there was significant reduction in the increase in dermal thickness were obtained from 12 hours to 48 hours. And these significant reductions were significantly ameliorated in the groups which were concurrently exposed with the Phyllanthus neruri and Acantha's aspera. Now, this table depicts the effect of daily oral administration of the acifate on increase in skin thickness after post-challenge with the DNCB dye. In this also, there is no significant difference at day 0, while there were significant differences, significant reductions in the increase in dermal thickness in the treatment group from 12 hours to 48 hours. And this reduction was significantly effectively ameliorated in the warts which were concurrently exposed with the Phyllanthus neruri and Acantha's aspera. And there was non-significant increase in the dermal thickness in the warts which are from the group treatment 1, means these are the warts which received least concentration of the acifate. Now, this table depicting the delay type hypersensitivity response to phytohemoctolin A. Following daily oral administration of the acifate, in this also there is significant reductions in the group T2 and T3 after 6 hour interval, while it was significantly reduced in the all treatment group, means T1, T2 and T3, which were effectively ameliorated in the warts which were concurrently exposed with the alcoholic extract of the Acantha's aspera and Phyllanthus neruri. Now, this is showing the effect of daily oral administration of acifate on splenic lymphocyte proliferation. Stimulation after stimulation with the mitogen concanavalin A, that is used for T lymphocyte proliferation. In this, there is significant reduction in the stimulation index in the warts which are from the treatment group as compared to the control, while this reduction in the stimulation index were significantly ameliorated in the groups which were concurrently exposed for the Acantha's aspera and Phyllanthus neruri. Now, this is the table that depicting the effect of the daily oral administration of acifate on splenic lymphocyte proliferation after stimulation with the lipopolysaccharide. This is used for the B lymphocyte proliferation. In this case also, there was significant reductions in the stimulation index in the treatment group as compared to the controls, and which was effectively ameliorated when they were concurrently exposed for the Phyllanthus neruri and Acantha's aspera. Now, this is table which is again showing the stimulation index against the RDF strain. In this case also, there was significant reductions in the stimulation index in the treatment group as compared to control. Now, this is showing the effect of the acifate on the absolute organ weights. So, the organ weights of the liver, spleen and thymus as well as the vasa fabriceus were significantly reduced in the treatment group as compared to control, while there was a little increase in the liver body weight that was not significantly increased while it was significantly increased in the spleen body weight in the groups which are from the least exposure of the acephate. Now this shows the effect of daily oral administration of acephate on the relative organ body weights. So in this case also, there was significant reduction in the relative weights of the lymphoid organs, especially in the wards which are from the treatment group and which have received the highest, extremely highest concentration of the acephate, and these reductions were ameliorated with when these wards were concurrently exposed for the Phyllanthus nereuri and Acaranthus aspera. Now this is the histopathology of these sacrificed birds, and this is showing the fibrosis in the Bursula fabriceus and depletion of the lymphocyte. And this is the photomicrograph of the spleen, which is showing the hemorrhage as well the lymphocyte depletion. And these lymphocyte depletions were significantly ameliorated when they have been exposed with the Acaranthus aspera and Phyllanthus nereuri. In the last, I would like to conclude that the subchronic acephate exposure at experimental low toxic dose may act as the immunostimulator in the experimental chicks, while the subchronic acephate exposure at higher concentration may suppress immune responses in avian species, and the concurrent exposure of Acaranthus aspera and the Phyllanthus nereuri may significantly ameliorate the immunotoxicity caused by subchronic acephate exposure. The bioactive molecules from these medicinal plants may facilitate the herbal drug development for preventing and treating the human and animal diseases. In the last, I would like to conclude. I would like to acknowledge the HOD of Pathology, Microbiology, Biochemistry and Data Animal Biotechnology Center, Nanaji Deshmukh Veterinary Science University, Jabalpur, India, for their constant support, encouragement and scholarship guidance, as well as to Dr. M. Thakur, who provided the technical guide of the acephate. Thank you.
Thank you, everybody.
Okay, thank you. I wanted to ask you about the white leghorn cockerels birds in your Indian country.
Yeah.
Is it domestic used and eatable birds?
Yeah.
Especially
these birds have been procured from the Phoenix group of the poultry farm, and
yes,
that is being used hugely in India by the Indians.
Yes, it is cooked.
Yeah.
So you do this experiment to show if there is a toxic hazards by consuming this type of birds?
Actually, I have conducted this research to identify
the immunotoxic.
As you know that this compound acephate is the least toxic,
yes,
least toxic organophosphate compound. Generally, it is being reported by the companies that that is the least toxic. So the farmers are using in a huge amount to increase their agriculture productions.
Yes.
But as they are using this is least toxic, so this is enormously used, and this is leaching out into the water and feed, and ultimately it is reaching to the non-target organisms just like the human being and animals.
So these birds exposed to environmental toxin by these pesticides in the farms in India by inhalation or by ingestion of this?
By ingestion of the feed and water.
Feed and water. Okay, thank you.
Any other questions?
Apparently not. So, thank you. Because of your,
Thank you
in this
very
case, no, we should announce one.
Yes, we have to announce
change.
one change to the subject. Is Dr. Tilak here? Okay,
So
Dr.
in this
Tilak
case,
is not here.
yeah. So in this case, we will move to the last presentation
Dr.
that it has been
Olujimi.
scheduled for this afternoon.
Dr. Olujimi.
if you're looking at him, it has been scheduled for today from 15:50 to 16:10.
Good afternoon, everyone.
Can you use the microphone, please?
Yeah.
Thank you.
Good afternoon, everyone. My name is Olanrewaju Olujimi. I'm from Federal University of Agriculture.
Can you hear him?
Can you hear me?
No, I don't think so. Can you go close?
Okay. I'm from Federal University of Agriculture, Abeokuta, and I'll be presenting on concentration, distribution, and health risk assessment of phthalate esters from wastewater streams.
No, I think they
from wastewater stream in Lagos and Ogun State. And this project is just a continuation of a study that I did in South Africa on looking at the effectiveness of wastewater treatment plants at removing some selected endocrine disrupting chemicals, which involves method development, analytical method development for phthalate esters and phenols in water. But for this particular study, which is sponsored by International Foundation for Science, Sweden, we to look at the effectiveness of oxidation pond and locally operated wastewater treatment plants. As we all know that water is very vital for life, and African is most countries in Africa are water scarce, and where there is water, people travel miles to get this water, which are not actually, in most cases, meet the international standards. So water has been looked at as the fresh water, to be precise, as a small fraction of available water which can be consumed to meet daily needs. And in most rural African countries, in more rural settings, you see women traveling miles of kilometers to get potable water. And in urban centers also recently, there has been water challenges, potable water challenges, coupled with poor management of solid waste management in cities like Lagos, Abeokuta, Port Harcourt, Kano, Kaduna, Abuja. So, and when you look at water quality, it's very important, since most people tend to provide their own water source or make available water for themselves to consume. So this study is actually on looking at wastewater management vis-a-vis water utilization for urban irrigation. Most people that do what we call Fadama agriculture. It's a World Bank funded project also in Nigeria. So it's been established that you have a lot of chemicals now, organic pollutants being released into water bodies, either from poor landfill management, because mostly in Africa now, waste are not actually properly managed. You have open dump everywhere, and by the time there is rainfall, you have a production of leachate that leads to groundwater pollution, and subsequently to water or stream around. So the quality of water is going down as the population increases in most of the urban cities. So the major objectives, it's not moving again, I'm going to need help.
Sorry?
It's not reflecting on the board, on the projector. But just moving on here. It's not working. No, it's not working. It's supposed to move down. I see the material and method is not coming up.
Okay.
problem?
I don't know, it's again the same problem. Doesn't move this whole screen. Apparently it's working now.
The major objectives of the broad objective of this study was actually to look at the efficiency of two
Please, can you use the microphone?
two wastewater treatment processes. That is, we have the oxidation pond, and the other one is the mechanized sort of mechanized system. And we intend to look at the physicochemical parameters of water in terms of how effective are these two systems. Are they able to actually remove these compounds? Thallic ester is an endocrine disrupting chemical which have been found in plastics and the bottles that they get because they are not actually chemically bounded, they are covalently bounded, they are easily released into the environment. And they are proven to be carcinogenic. For example, DHP, diethyl, and zitalics. And we also intend to assess the probable effects that this can cause in children or adults when they get exposed to contaminated water, especially people in rural areas or even some urban centers that don't have access to potable water from government source. We have two sites. As I said, we have one oxidation pond which was within a university community, a private university community, and the other one is a government-operated water treatment plant, which is the first one here is a wastewater treatment plant at Ikeja. Ikeja is the capital of Lagos. That is the most popular city in Nigeria. There are 17 million people. And the treatment plant was constructed in 1979. And for the small one, it's not able to meet the challenges of the population today because the population has actually grown. And the other treatment process is at Covenant University in Ogun State. So it's an oxidation wetland system kind of a thing that was constructed for water to move. No special treatment processes, just natural processes. So for our method, based on the method that I developed earlier in 2011, the same method was used for little modification. In that methodology, what I did was to use SPE method, but in this analysis, I use a liquid, which is very common and very simple to use. Because we're dealing with solids, we make sure we make use of plastic materials because they are easily mixed into the water. So what we did was glass also. The glass were also cleaned using standard procedures, and they were baked to eliminate solids because one can easily come in contact with solids. Our water samples from this mils of water sample were collected and extracted thrice using 40 mils of DCM. These were combined together and evaporated in a field cupboard. And the normal procedure of cleaning was followed. That is the silica gel cleaning. The silica gel was activated in a field in an oven at about 250 degrees for about 16 hours. Then the water parameters, the physical and chemical parameters were analyzed. The COD, BOD, the normal pH, we have the in-situ and the ex-situ. Yes, 4 minutes. Then the risk assessment. For the risk assessment, we only consider two parts to it. That is the injection and the dermal contact. The injection is for people that might actually come in contact with water downstream. You know, we have the influent, we have the effluent water, and once the effluent water is released into the river, into the water body, villages adjoining the water course can actually make use of the water for their domestic purposes. And behind the Covenant of Freedom Fund that is behind the university, we have what we call the Fadama Farm. The Fadama Farm is the Fadama Farm is behind, and they use the water for irrigation to irrigate vegetables. And the irrigation is that the vegetable will be harvested and taken to the market for people to buy and use. And on the risk assessment, the USEPA method was the model, the development model was used. It had a lot of computations and just speak through. But two things I want you to note is the cancer risk assessment and the non-cancer risk assessment. For the cancer risk assessment, the acceptable limit is between 10 raised to the power minus 4 or 10 raised to the power minus 6. So you can say somebody can actually develop cancer if your values are actually below this. There is potential of people developing cancer. And the non-cancer effect, the HR, which is the hazard index. Once you do your summation and it's up to 1, then there will be non-cancer effect. Right. For our studies generally, the values were actually above the FEPA standard. FEPA is a federal environmental protection agency in Nigeria. We have standard limits for all this. As you can see, most of the values were actually above, except the DO from the study size. In terms of our recovery, to be sure that our method that we developed was okay in terms of extraction, the range was between 83 to 98.4%, which is quite okay. Then the mean talent concentration for the study period in the raw was 151 microgram per liter, which is high. The acceptable limit for pellets in water is 3 microgram per liter. This is far beyond what is acceptable. And looking at the variation in influence, what we did was we compared the two wastewater streams, what comes into the streams and what goes out. What we discovered was that at Ikeja wastewater treatment plants, because of the population and the location, we have more inflow of pallets in the sites compared to the one because they don't know which service only the university communities. And this just shows the correlation in terms of what we're trying to see is the source apportionment. That's what we're trying to do here. Okay. Then in terms of percentage distribution, the distribution pattern, there's a kind of variation between the two sides. At Ikeja, we have BEHP was common, But DNP, which is dimethylphthalate, was more prominent at Ikeja than at Covenant than at Ikeja. And this is because DNP is widely used in shampoo, and it's a small community, and is the oxygen pond only receive waste from the community, from the university community, while the other one receive waste from both industry and the people in the domestic wastewater. In terms of percentage removal, on an average for the study period, Covenant Oxygen Pond was able to remove 43.3%, while the wastewater network was able to remove 54%. This was far below what we actually discovered in South Africa. Way below, because in South Africa we have sometimes 100% removal of targets, and this just shows the performance level of the two systems. And looking at the carcinogenic and non-carcinogenic waste, from the study, we discovered that children are more prone to develop cancer than adults, and that is what is expected. This is just showing the overall index for the two sites. And in conclusion, as I said earlier, DEHP was found to be most abundant, which is what has been widely reported. But in the recent study that we carried out in dump site in Lagos, which is being funded by Lagos State Government and World Bank, we are having a different total pattern of distribution. That is going to be for another day. And finally, we said children and adults are likely to develop cancer risk due to level of DDH. These are some of the selected references. Thank you for listening.
Yes, please.
Okay. Thank you so much. I visited Nigeria and I go to Lagos and the Kupa. And I wanted to ask you about why is there is a high level of pollutant PES in February per se. I don't know. I wanted to know.
Yes, that's what I'm saying. Because we can say this is what is actually responsible. It could be from the production line.
From industrial production line? Yes. Okay.
It could be from there because in Lagos we have industry releasing their waste to the site.
So it is in February more in Lagos?
More in Lagos.
Yes. Okay. Thank you.
All right. Thank you very much. Would the presenters please come up so that we can present the certificates?
Yes, I think so.
Yes.
You're okay.
Okay. Thank you.
Everyone that was in this session.
I think you can sign this for me.
Yes.
Our first speaker, which you well know, this is Professor Diana Anderson, who will present a paper entitled The Protective Effect of.
Very much. Right, this is a follow-on really from what I talked about yesterday, because I introduced a lot of concepts there. If I can just go through this.
Let's see what I can do. Oh.
Thank you very much. Okay. Now, this is, as was explained, the protective effect of flavonoids on food mutagen-induced DNA damage. And again, I'm using peripheral blood lymphocytes from cancer patients. And here I have a colleague whom I've known for some time. Yeah, I'm going to tell you, he's got a second life, which he actually does travel books. Right. Okay. Now, the names of the food mutagens, which I could not remember from the top of my head yesterday, were IQ, and this is 2-amino-3-methyl imidazo 4,5-quinoline, and FIP, which is 2-amino-1-methyl 6-phenyl imidazo 4,5-pyridine. And these are heterocyclic amines, and they're generated when heating proteinaceous food. So food itself can produce potentially harmful products. So I think I said this, if there's not something in the body to protect you, you could be mutated out of existence. Of course we're not, we're all here to tell the tale. So something is protecting us. Now, this study investigates the protective potential of the flavonoids quercetin, which is called Q here, and rutin, R, against oxidative stress induced in vitro by IQ and FIP in lymphocytes from healthy individuals and untreated, newly diagnosed colon cancer patients using the COMET assay. And you can get quite a lot of information out of these studies using lymphocytes. Colorectal tissue is constantly exposed to different chemicals and free oxygen radicals formed during metabolic activation. And the inflammatory bowel disease, which some people have, is associated with increased risk for colorectal cancer. And so are various lifestyle factors, and these include cooking-derived carcinogens, such as the heterocyclic amines. Now, the daily intake of the heterocyclic amines can reach 50 micrograms, depending on the meat type that you eat, the cooking method, and the temperatures used. HCA formation seen as heat-induced non-enzyme browning involving creatinine, free amino acids, and monosaccharides. And we actually know that FIP induces tumors in rats. And IQ is classified by IARC, which is the International Agency for Research into Cancer, as a 2A category, and FIP as a 2B. So they're on the books as quite powerful chemicals which could eventually be classified as human carcinogens. HCA, heterocyclic amine genotoxicity, originates from reactions involving cytochrome P450 when the parent compound is converted to an electrophilic derivative such as a nitrinium ion covalently binding to DNA, and this results in adducts and nucleotide alterations. And another major causal factor, obviously, we've talked about it so much at this meeting, is oxidative stress. I feel oxidative stress is just everywhere because, as we were talking, we've talked a lot about estrogens too, and I mentioned that yesterday. And estrogens do have oxygen radical involvement. So free radical scavenging activities are found for several kinds of abundant low molecular weight polyphenolic compounds called flavonoids, and these are present in fruit and vegetables. They can act as pro-oxidant or antioxidant depending on their concentration. And we've had talks here where they've acted in the right way as antioxidants, but you mustn't forget they can switch responses and go the other way. And in 1997, we observed positive responses with flavonoids in the COMET assay when the lymphocytes were treated alone. But in combination with the food mutagens, they show exacerbating effects at low doses and can be protective at high doses. So there's always complications somehow. Now, how do we get these samples? Well, we have informed consent, and 10 mils of heparinized blood were taken by venipuncture from healthy volunteers and 20 colon cancer patients from the Department of Gastroenterology at Bradford Royal Infirmary. This is the hospital which is just across the road from us. So it's really very lucky that we have access to patients and that the consultants have agreed. It takes ages to get ethical permission in the United Kingdom. I expect it's not true for everywhere, I know that. But it took us a year and a half to get this. I mean, that's a lot of time. If you've got a grant that's lasting 5 years, a year and a half to try and sort out the ethics is quite a long time. So you have to get that, and then you also have to get consent from normal healthy individuals. And we use the normal health of individuals who come from the university. And these are made up of staff and students, because you've got to have the right matching age ranges. Over a period of 2 years, samples were collected, and the age range was between 18 and 90, with a mean of 50 years, plus or minus 19.8. And they were grouped in intervals of 18 to 49 and 50 to 90 years. And we grouped them like that because we wanted to consider their confounding properties. Confounding factor data collected involved endogenous information such as age and gender, and exogenous alcohol, smoking, diet, and medicine intake. And also in Bradford, as I said, we have a lot of Pakistani population, so we have ethnicity as well. And the chemicals were bought from standard suppliers such as Sigma. Cell viability, you have to have good cell viability. If you don't get suitable cell viability and your survival is usually over 80 or 90, then you can't tell whether the effect that you're looking at is due to genotoxicity or cytotoxicity. So we always keep it out of the way. We always keep high, that's why we measure the actual numbers of healthy cells. Lymphocyte suspensions, 100 microliters of 10 to the 6 cells per mil, were exposed to defined concentrations of food mutagens and or flavonoids in the presence of the medium, RPMI, and a total volume of 1 mil. And we use different concentrations of IQ and FIP. And the IQ, we worked this out beforehand, the doses that we should take to avoid toxicity, and FIP also. And for the modulatory effects of flavonoids, the highest concentrations of IQ and FIP were simultaneously combined with quercetin and rutin. Again, that's a non-toxic, these are non-toxic concentrations, 50 for rutin and 250 and 500 micromolar. And the COMET assay was the primary assay used in this study. Now, you saw these yesterday, just to remind you, those of you who don't know the COMET assay, but here you can see an undamaged cell. Here you can see quite a damaged cell. You probably can't see this, but it probably goes out to about there in terms of damage. And this damage is due to the DNA moving towards the anode. Here's one that is less damaged. But you can, as I said, if you're living in a country where you can't get access to this, you can make your own scoring up, provided it's standardized, and you can just do it without a computer. We're doing this with a computer. But you could just say this was a high damage. Just give an example, high damage, low damage, no damage. So you can do it, you know, as we all used to before this became popular. And that's what it looks like. And here, again, you can see these moving towards the anode, the damaged DNA. It's put in lysine solution to unwind it, and then into electrophoresis solution, and the migration of the DNA during electrophoresis. And you see the microslide, and you've got first layer of no melting point agros, and then low melting point agros in which the cells are damaged and undamaged cells are embedded. And then the third layer of agros on top of that, a cover slip. Now, there are two ways of showing the data. There's percentage tail DNA, which indicates the DNA damage in the lymphocytes after in vitro treatments with IQ and FIP with either the flavonoid quercetin or rutin, and in healthy individuals and colon cancer patients. I'll show you that in a moment. That'll be figure 1. And then DNA damage induced in vitro in lymphocytes from healthy individuals and colon cancer patients by the food mutagens, IQ and FIP, and its reduction by flavonoid supplementation. Various concentrations of Q and R, that's in table 1, and confounding factors for healthy individuals and colon cancer and their influence on the baseline DNA damage using the Comet assay, that's 2. So let's move into figure 1. Now, here in A, well, I'm going to talk about the piece at the bottom. We've used percentage tail DNA, or we could equally have used the excuse me, olive tail moment, which measures not only the length but the density. The Comet parameter percentage tail DNA is shown indicating DNA damage in lymphocytes after in vitro treatment. Excuse me. If you're not used to air conditioning, which unfortunately Brits aren't, when you come into an air-conditioned room, it catches you. You don't know how lucky you are to be in air-conditioned rooms. We live in a country where we're constantly at low temperatures. Okay, now let's have a look at A. Now before we do that, just go back onto this. We've got 4 panels here, and in panel we have IQ and R. Let's look at the first panel A. You can see here, this is the healthy individual, and these are the colon cancer patients. And it's the same in all of them. And in this one, it's you have Q, and in this one we have R. And this one again we have Q, and this one we have R. Now what are we trying to find out here? Well, I think you can see, let's just take this example, 0, 25, 75, and 100, and up to 150, and then we take this one at 150, and we get the values from the IQ supplemented with Q. Now, I think it's quite clear from all of these that the cancer patients have a different baseline. And that, again, was something that we've seen in all the studies that we've done. Cancer patients are higher than healthy individuals, and that was the basis of this test I was telling you about yesterday, where you can tell if a person has got a very much higher level than baseline, then they might be more susceptible to cancer. But this work was done before that, but we kept finding this. And here you go up, they both move up the dose-response curve, and then we apply the 150 micromole of IQ supplemented. And here you can see supplemented with the quercetin, and again, they come down. But what you will notice is that the cancer patients do not come down as well as the healthy individuals. So this is other evidence of the difference between the cancer patients. This is the same here, where we are supplemented with rutin. And here the cancer patients are higher, and they come down. They don't come down as far as the healthy individuals. Here we've got treatment with FIP, which is the other food mutagen. Again, there's a difference between when you have the error bars. You can see a distinct difference. There's 20 individuals in each group here. Now, as you think, if this was an animal study, you would not be allowed to put 20 individuals in today. You'd have to be down to about 7, I think, in guidelines. But that's a lot in terms of looking at these sort of things. That's a lot of individuals. And this shows exactly the same pattern with FIP. Doesn't quite get back to the values of the control. And again, in D, this is where we've treated with FIP, grade 2 carcinogen. And here we get this difference. So they're showing exactly the same sorts of patterns. And this is what could happen. This is happening in vitro, but it could happen in vivo. Now, there's 20 individuals in each of these. There's quite a lot of observations here as well. We've been amassing this evidence of the difference between the disease state individuals and the healthy individuals. I'll walk you through this because the numbers, I don't know if you can see them all. But here we have our negative control. That was the value that we were having here, 2.1 plus or minus 0.3. And then you give these IQ, the food mutagen, and you can see how the responses increase from 3.23 up to 8.47. Highly statistically significantly different. And here you can put it in percentage terms, how this increases. That was 100%, that will go up to 410. Now for the colon cancer patients, they start off at this higher value, 4.10, go up in a dose-related manner as you go up that dose-response curve. And again, if you put it in percentage terms, you go from 100 to 237. Now, what if you add the IQ and the quercetin? And what happens there? Taking that highest dose, what happens? You can see in numerical terms that it comes down from 7.67 to 3.44, and from 366 here down to 1.43. If you add rutin, 7.36 to 4.12, and the value here is 3.69 to 1.96. And so we go across for each group in that way, healthy individuals behaving in the same way here, and then the colon cancer patients, how it is affected there. These are for each of the groups. I'm just showing you the individual data. Now we're on to FIP and quercetin and FIP and rutin, following the same patterns for the colon cancer patients and the healthy individuals, the colon cancer patients there. So DNA damage induced in vitro in lymphocytes from healthy individuals and colon cancer patients by the food mutagens IQ and FIP, and its reduction by flavonoid supplementation with various concentrations of quercetin and rutin. Now, when you think that these are actually in the fruits we eat, so we're very lucky that we do have things like this in the fruits that we eat to prevent these effects that we don't want. Do you know that some people can't eat fruit or have an allergy to it? Now, what does all this tell us, and what have we got to do with all these confounding effects? So you have to, in all studies like this, have a good statistician who can help you. And here we've got the healthy and the colon cancer patients and the values. And colon cancer patients versus healthy individuals. And you can see the differences there. 2.10, 4.10. And this is when you look at them with these different factors. And here's the age. When I told you we split them up into two age groups, greater than 50 and less than 50. And if you look at those, over 50 years gives you more damage. So why do we age? Here's some evidence for it. And what about the smoking? We all know about smoking. Regardless of the fact that we do know about it, people still smoke. And I once worked in a cancer research institute where the highest smokers that there were found there, actually. And what you can see here is the active smokers 3.16, ex-smokers 2.69, and non-smokers. That wasn't much different there, even though they could have smoked a long time ago. And the active and ex-smokers versus non-smokers. What about drinking? And as I say, we have people in our population who don't drink, or if they do, I don't know, who say they don't, their religion doesn't allow them to drink. Now, severe drinkers have higher values of, here, 3.20, moderate drinkers here, and no alcohol here. So there we've compared all that. The diet, Western diet, which is what we've been eating today, most of us, Asian or vegetarian, then you can see that there's an effect of, that says to us that all Asians should be living longer, perhaps, but it doesn't always work out like that. So Western versus vegetarian diet. Gender, and is the male better than the female or vice versa? Well, the female has lower damage than the male. And that fits our lifestyle. Generally, females live longer than the males. This takes its toll, all these things, drinking, eating, working, everything. So when you split it down like this, it all makes sense. You've no idea which way it's going to come out until it's come out. So then there was the point, right, well, do any of these affect our data? And the answer was it wasn't confounding enough to affect the data. But you've got to go through this process of doing it. Now, heterocyclic amines are created when cooking meat and fish, and food mutagens which pose a potential risk to humans. Strong evidence that endogenous liver and kidney enzymes or commensal bacteria in the intestine, there was a question about the intestine this morning, and the intestine are able to metabolically activate heterocyclic amines, which in turn generate DNA damage. In 2004, Poul Sobel, she died actually not long ago, found lymphocyte responses to be very similar to responses in rectal cells, thus supporting their role as surrogate cells for biomonitoring. Because this question comes up all the time, why are you using lymphocytes? And well, it actually does. They behave in the same way as other cells in the body, which is not surprising when you think about it, because your cells in your body are the same for every cell that you have. And I've said this before, according to IARC, IQ is probably carcinogenic to humans, and FIP is possibly carcinogenic to humans. And that, number 1 is a human carcinogen, 2A is probably, and 2B is possibly carcinogenic. Strong antioxidant effects of flavonoids to protect against DNA damage are known. And we've been working in this sort of area for many years. Statistically significant responses for food mutagens have been shown in lymphocytes in our studies when measuring both olive tail moment and percentage tail DNA. The thing to remember, baseline damage is higher in cancer patients in this, and has been shown in subsequent studies, was definitely shown in our 2014 paper. The modulating effects of flavonoids in a co-treatment with a high dose of food mutagen seem to be affected by higher baseline damage levels, as damage from cancer patients was not reduced to damage levels in the healthy individuals. And I do believe that we are born that way. I think that there are 1 in 3 people get cancer at the moment. They say it's going to be, by 2027, it's going to be 1 in 2. What is making that rise? Is it because we're living longer or whatever? But that means half of us or a third of us in the room are going to die of cancer. We have to die of something, so I don't know if philosophical being like that or not. Or perhaps you don't want to know about it, you can just live and see what you die of. At least you won't know what you die of, but they operate through, flavonoids operate through antioxidant properties. Flavonoids are able to effectively protect the integrity of lymphocytes' DNA from reactive oxygen species. Therefore, an ideal supplement compound to prevent the onset of cancer. But if you eat, do you say in the States, eat 5 pieces of fruit a day? Yes. That's what they're always on about in Britain. Eat 5 pieces of fruit, and that will help. You get it in Slovenia?
Yes, yes, yeah. Also in Slovenia.
Not striking a chord with the others of you, so it doesn't look like you. It is 5 pieces of fruit a day. And if you don't like fruit, then that's tough. But there we are. Thank you very much indeed.
So thank you very much for a very interesting presentation. Any questions?
Can you tell me what this baseline DNA damage being higher in the cancer patients? What is that just explained by the fact that these patients are taking chemotherapeutic
baseline
agents?
Damage, right, is lower than in cancer patients. So if you're a cancer patient, you start off with higher damage. So if we take a cancer patient and take you, and you're a healthy individual, I mean, I'm talking in general now,
yeah.
Then on the whole, out of all the people we've looked at over the years, we have found that cancer patients generally have a higher starting point. Now, it's based on that premise that we're saying that that person might be prone to cancer. And it's because of that test I showed you yesterday, where, because I didn't explain specificity and sensitivity to, but does anybody not know what specificity and sensitivity is? Well, in a nutshell,
we all know.
If you're going to, any good test for cancer should be able to tell you whether, in fact, you'd be able to pick up the damage. You'd have a high sensitivity. Whereas if you're not going to get it, you'd have a high specificity. Mm-hmm. You know that? Yeah.
No, yeah, but I guess what my question is, when you say cancer patient,
yeah.
Do you mean a patient that is already taking chemo?
No, these people that we're looking at, they've not had any treatment whatsoever.
Okay.
So, yes, yes. They've had no treatment, but I believe that those people have got it. You're born with this. Some people are going to get it, whatever. Because I'm not, the tests that are out there, that the British government, that the American government have spent millions on, most countries around the world, are looking at genes. What is, right, it's great to know that certain genes are associated with something, but what is the point of looking at hundreds of genes? You still don't know if you're going to get cancer. Whereas if you look at the whole genome, which is what we're looking at here, it's a simple, very simple COMET assay, looks at damage to the DNA. It's like cracking a nut with a hammer. And then
when you did your study, when you got your samples, you got the samples from cancer patients from the hospital, right? So those patients already had cancer. You didn't actually take a sample before the diagnosis was made.
They had it, right? They didn't know that they had it, but they went to the doctor saying, I've got certain symptoms. So the doctor then sends them to a clinic, and the specialist at the clinic checks out various things and says, at that point we get them, by the way, we get them, but they've not been treated.
Okay, all right, cool.
But in this case, for instance, if you pay attention to the organ...
Wait, I'm sorry, I can't hear you. Here comes the...
Yeah. In this case, with these patients, regardless if they have been diagnosed or not, it's a fact that they have more lesions in their DNA than the control ones.
Yes.
These lesions are single-strand break, maybe double-strand breaks, or alkali-labeled sites. Yeah.
Well, why is it that at this moment in time, 1 in 3 of us is going to get it? 2/3 of us are never going to get it. So what makes us, who never, I don't know if I'm never going to get it, but those who are never going to get it, what makes them different from those who do? And I think it's their sensitivity, their susceptibility.
To instability.
Yeah. Susceptibility to DNA damage.
Yeah.
And when you think about it like that, it becomes reduced to something very simple. Whereas you can do it by finding out that a person has 25 cancer genes, but it doesn't mean that they're going to get it. What means much more is that they're going to have this higher incidence.
I just wanted to know if you could comment on the age of the cancer patients, because you've mentioned...
The age, right. What we did, we age match. It doesn't mean we age match as well as we can. Because most cancer patients, if you think about it, well, the graph for cancer, when you're young, you often get leukemia, things like that. And then that sort of dies away in the 20s, and then you go gradually up. But by the time you're 60, you're getting on, and your enzyme systems aren't as efficient or as effective. And so we try to take people who are not over 60. You know, we take some who are, but we try to get a broad spectrum so that we can include everybody in that. I mean, it's very crude, what I'm talking about, but so far, it has proved very effective. And then these blind trials that we're now trying, we've done it with colon cancer patients. We don't know what we're getting. The person who's been doing these tests just gets something to test. We don't work, the consultants who give us the blood normally for research are not involved with us at this point. They're given to an independent tissue bank. We just get coded samples and then evaluated in that way. So everything is coded. We don't have a clue about what's happening, but it's seeming to work out at the moment. Now, in 2 years' time, when all this is finished, it might not work out like that. I don't know. I'm only telling you what seems to be happening at the moment.
So, thank you very much, Professor Anderson. And now I would like to introduce our next speaker. This is Dr. Balz Soweit, a pharmacist who obtained his PhD at the Free University in Berlin. And he will present a paper. Oh, silibidine. So we will change the order of presentation.
Phytopharmacon and its potential use for the treatment of sulfur mustard injuries. So what is sulfur mustard? It's a chemical warfare agent that has been repeatedly used in the 20th century. From World War I to the Iran-Iraq War. It has the very unfortunate track record of being the chemical warfare agent used most frequently in the 20th century. The case fatality rate is relatively low at 2%. However, it induces very severe symptoms, including skin blistering, ulceration, and inflammation. And after the acute phase with prolonged wound healing, leading to chronic injuries to eyes, to the respiratory tract, and the skin as well. Despite 80 years of biomedical research, there's no causative antidote known. It's not that we hadn't been doing any research anywhere in this world, but in fact the molecular pathology of sulfur mustard poisoning is highly complex, and it's perhaps impossible to find the single substance to treat it. However, the findings I'm going to present, I hope will contribute at least to an improvement of therapy. I apologize for this graphic images, but I think we have to face what we are dealing with. And even as we speak now, we have to consider sulfur mustard a threat. Some of the old stockpiles are probably still available in very unstable regions of this world. And even if all of those stockpiles were successfully disarmed, actually, the US-led effort in the last year successfully disarmed a lot of the Syrian chemical warfare stockpile, but we do not know what has not been handed over. And even if all the stockpiles had been successfully disarmed and destroyed, it's comparatively easy to synthesize with moderate knowledge and a moderate level of equipment. So this is what the acute injury looks like, and here's an example of the resulting chronic damage to the skin. In this case, hypo and hyperpigmentation closely associated. What is even more disturbing and more aggravating is the chronic injury to the respiratory system and the eye, often leading to permanent disability and severe impact on the quality of life. And in the long term, regrettably, we have to say also on life expectancy. So what can we do? Can a potential treatment perhaps be found from this plant? This is Silybum marianum, and from the seeds of the milk thistle we can extract a number of flavonoid compounds, but the lead compound is silymarin, also known as silibinin. It is already an approved drug, both for the treatment of unspecific liver damage, so liver damage arising from the abuse of alcohol or of the nutrient toxic effects, just an overabundance of food, of fat, of nutrients, but also as an antidote for the poisoning by Amanita phalloides. So this mushroom is also known as the green death cap. For a reason, poisoning with these mushrooms had a very high case fatality rate until silibinin was introduced into therapy. And in case of this acute and life-threatening emergency, we need to have a formulation and we need to have a prodrug to deliver it directly and systemically by infusion. So there's this water-soluble prodrug known as silibinin bisoxanate. Recently introduced by the work of Dr. Tiwari Singh from the Denver University, discussion was started whether silibinin might be a therapeutic compound to treat sulfur mustard injuries as well. She had been doing a number of really amazing studies on this, but because the University of Denver is a civilian university and this chemical is for very good reasons highly restricted, all the studies had been limited to simile substances, so nitrogen mustards, which are actually still in use for anti-neoplastic treatment. They had been used as the toxic challenge, and the studies had also involved free silibinin and not this soluble prodrug. So let's have a look at the genuine silibinin first, and highlighted here are two residues introduced of succinic acid, and these can be converted to their sodium salts, and we have a nicely water-soluble compound. The aim of our study was to investigate for protective effect of the water-soluble silibinin prodrug of SilBS, as we abbreviated it, the compound which is also approved for clinical use. We wanted to do this in our cell culture model and actually challenge this model with the real agent with sulfur mustard, and because the Bundeswehr the Bundeswehr Institute of Pharmacology and Toxicology, where I'd been working at until fairly recently, is part of the German military, of the military medical service. We have, under tight regulations, of course, access to very limited amounts of this compound. But as you will see in the cell culture system, only very tiny amounts of this are required to do some meaningful research. In the model, we wanted to assess both the overall cytotoxicity, the apoptosis, because apoptosis is a key mechanism in sulfur mustard injury, and the inflammation. We also wanted to know whether silibinin bisoxanate, the prodrug, could actually be transformed into free silibinin. So here's what we did. We started with a relatively simple model, no co-culture. I'll come to that in the second talk. But this, in fact, was a monoculture of immortalized keratinocytes known as the Hackett cells. Those were seeded in 96 well plates at a density of 50,000 cells per well. We supplement, we grew them in the Becker's minimum essential medium, supplemented by 5% of fetal calf serum, and exposed the cells to 30, 100, or 300 micromolar of sulfur mustard in Eagle's minimum essential medium. There was no serum supplementation during the exposure phase because if you would do this, a large fraction of the sulfur mustard would actually bind to the protein from the FCS, and you would obtain less reproducible results. 100 micromolar of sulfur mustard is a fairly well established exposure level in literature. That's often regarded as the under limit of exposure that will reduce in acute toxic effects, whereas 300 micromolar is already a severe exposure, and at least in the cell culture model, in otherwise untreated cells, induces, let's say, 90% cytotoxicity, apoptotic or necrotic cell death within 24 hours. We treated those cells either under sham conditions, of course, or with 10, 50, or 100 micromolar of silibinine bisuccinate. We actually calculated these concentration levels from the pharmacokinetic data we have. So the pharmacokinetic literature says that during acute and intensive treatment for patients with Amanita phalloides poisoning, 50 to 100 micromolar silibinine bisuccinate is the clinical level you actually achieve. So to the left side, you see these Hackett cells under control conditions, a very nice and regular and very dense pattern of cells. You see, after 300 micromolar challenge with sulfur mustard and a 24-hour post-exposure incubation time, there's largely cell debris, very few cells alive anymore, and even those that may remain intact already in apoptotic conditions. On the image down below, you can see our high throughput ELISA system. So both me and my PhD student, we were quite grateful for having an automated pipetting system capable to operate a sort of high throughput system. We did all our assays both for the necrosis, apoptosis, and the inflammation on the automated pipetting. So start with the necrosis assay first. We used the Toxylite bioassay to determine the level of adenylate kinase both in the supernatant and in the lysate of the remaining cells. Now, adenylate kinase is an enzyme that appears everywhere in human cells, but it should not appear outside the cell unless the membrane integrity is lost and the cell is in the status of necrosis or aponecrosis. We then calculated the ratio of supernatant AK versus total AK, and we came up with a fairly good assessment of the percentage of necrotic cells. The apoptotic activity was assessed with the cell death detection ELISA. You actually detect the nucleosomes from the cell lysates, and those mononucleosomes, they are highly specific apoptotic markers. We also used the cell count or a surrogate parameter for the cell count, the AK levels from the lysate to normalize. and to get a fairly accurate impression of the apoptotic activity. Interleukin 6 and 8 as the inflammation markers, they were assayed from the supernatant and using standard ELISA procedures. In a very separate experiment, we cultured flasks of Hackett cells, cultivated them with 100 micromolar of silibinin bisphosphonates, collected the supernatant, lysed the cells, and there was no sulfur mustard involved in this experiment. We just focused on the capability to transform the prodrug into free silibinin, and we analyzed both fractions using an LC-MS system. So now on to the results. As we can see, fortunately, there's a significant reduction of necrosis using 50 to 100 micromolar of silibinin bisphosphonates. This is particularly impressive in the after subclinical exposure and low-dose clinical exposure. So these are not clinical exposures, of course, but the challenge is similar to what would result in acute clinical symptoms. And even under severe exposure conditions, we found a small but still significant reduction of necrosis. And until that day, we had never really found another compound capable to reduce the overdose, especially in post-treatment. There are a number of compounds that you could possibly use for pre-treatment, but let's face it, you're never really going to know when this kind of poisoning is going to affect you. And if you were aware of the risk, you would probably take means of physical protections like a respirator or protective suit rather than pre-medication. So it was highly valuable for us to actually come up with these findings that protective effects could be achieved by post-exposure treatment. On to apoptosis. We found very little under the low and medium dose exposure, and even a slightly, but very slightly amplified apoptotic activity in one single experimental group, referring to the group that had been exposed to the highest sulfur mustard challenge and the highest dose of subsequent silibinin treatment. Regarding inflammation, IL-6 production can also be amplified by high-dose treatment with silibinin. And the effect on IL-8 production is somewhat ambiguous. So initially, looking at the low dose and medium dose exposure and the subsequent treatment, you see that a high dose challenge, medium dose challenge, but high dose silibinin treatment, you see there's a significant reduction of the IL-8 production. But if you go towards these severely exposed cells exposed to 300 micromolar sulfur mustard, and you add the high-dose silibinin treatment, the effect is turned the other way around. There's a slight but significant amplification of IL-8 production. Referring to the biotransformation, here's a chromatogram we had obtained from the lysate. And as you can see, we have nicely separated peaks, both of silibinin and silibinin bisphosphonates and the internal standard. we could obtain psilocybin mass spectra to unambiguously identify our substances. And our findings were that we found peaks for free psilocybin and psilocybin bisuccinate and monosuccinate. The molecular weights and fragmentation patterns were all very much in agreement for an unambiguous identification. Psilocybin bisuccinate was dominant in the supernatant, and traces of free psilocybin and psilocybin monosuccinate, so to say the intermediate cleavage product when just one succinyl residue is removed, they were found in the supernatant. And surprisingly, in the lysate, psilocybin itself, the free psilocybin formed the largest peak. Psilocybin bisuccinate, the prodrug, was the smaller peak, but no psilocybin monosuccinate was found. And we concluded that the prodrug could be absorbed into the cells. And within the cells, we had actually a cleavage of both the bisuccinate and the monosuccinate ultimately to the free compound. And probably as a result of the monosuccinate cleavage happening at a higher frequency, the monosuccinate totally disappearing from the lysate fraction and the free compound forming the dominant peak. In summary, we have a significant and dose-dependent reduction of necrosis, and these results could be obtained after challenge with blistering, so actually clinically relevant sulfur mustard concentrations, and also at clinically relevant concentrations of psilocybin and the prodrug. Regarding the post-exposure treatment that had thus been a unique protective effect among the substances tested at this time. There was a slight but significant but probably clinically irrelevant increase of apoptosis only in the severely poisoned cells and only under high-dose treatment. This was largely in agreement with our finding in literature that in cells which are already under DNA stress, so including malignant cells, their findings also with regard to pro-apoptotic effects of psilocybin. There are some studies which have even tried psilocybin as a potential supportive treatment for patients with liver cancer using these pro-apoptotic effects. The increase of inflammation under high-dose treatment with sulfur mustard and psilocybin bisuccinate can be explained by a survival of severely poisoned cells. So we rescue those severely poisoned cells in the first place, but the consequence is the surviving cells reward us, so to say, by a cytokine storm. They are still severely damaged. We can save them for the time being, but we cannot reduce their inflammatory activity. In summary, due to the complex molecular pathogenesis of sulfur mustard injury, psilocybin will not be the miracle drug. It will require multiple interventions, multi-drug treatment, but psilocybin bisuccinate may well have a role in this because of its anti-necrotic effects. This could potentially be augmented by compounds to rein in the excessive inflammation. I'll present more on this in the subsequent talk, and also on the excessive extrinsic apoptosis. the slight pro-apoptotic effects might in the end be clinically useful to debride of irreversible DNA-damaged cells, to get rid of dysfunctional cells or cells that may turn into precursors for cancer at a later stage in time. Sildenafil bisoxanate has a very favorable regulatory status. It's already an approved medicinal product. It's known to be very safe and non-toxic. And our onward work should focus to identify the protective mechanism. So is it rather membrane stabilization? Is it an antioxidative effect? If you read into the sildenafil literature, you read about pleiotropic effects. So there are a number of protective mechanisms. We have to focus on dose finding. Maybe the 100 micromolar treatment is more effective, but the 50 micromolar possibly safer, avoiding pro-inflammatory effects. And we are going to test combined treatments in co-culture models. Thank you very much.
Thank you very much.
I would just like to make a recommendation about questions. We're already running a half an hour behind. Would you all mind if we held questions for our networking and posters? Would you mind if we did that? Just hold your questions and we'll, when we're networking and looking at posters, we can visit with each other. Is that okay?
Very much okay.
Thank you very much. And now our next speaker is Dr. Ledita de Freitas Talarico from University of Sao Paulo in Brazil, and she will present the relevance of assays using mollusks and perspectives in environmental risk assessment.
There's a tremendous bell.
Yes. I didn't know it was so, was it allowed? It was too. I am the next speaker.
I must you say.
Yes.
You can get it.
Okay. Hello. Good afternoon. I'm from Brazil, and I'm talk about here. I'm talk about a review about mollusks. I work with mollusks, specifically with snails, and we have some important species in this. This specifically work in environmental assessment, okay? We talk about the chemical compounds in aquatic environmental, cause effects and organisms interacting with natural stressors affecting reproduction, surviving, exposed population. And yesterday, Dr. Marcelo talked about the importance of new methods that organisms to replace, to reduce, refine the animal use. And here I explain in this way, explain the importance to consider new tools for understanding the relationship between the levels of contaminants in the ecosystem and their effects on the local biota. Because we have some species specifically for some countries, and some studies in ecotoxicology protocols use specifically some studies. Most used assays are acute and chronic tests, but nowadays we try to assess the potential sublethal hazards to ecosystems in low concentrations, not the only toxicity, acute toxicity to lethal effects, but effects in low concentrations. So the improvement of monitoring programs have encouraged the analysis effects in gametes, fertilization, reproduction, embryo-larval development. In this way, I can put for discussion the mollusks, because they are the second largest group in kingdom Animalia. But this group, they're not being considered in environmental risk assessment so far, because mainly due to the lack of the standardized protocols. But we have a lot of studies with gastropods in my country and in other countries. I show for you. Gastropods in the habitat is most abundant and with great importance in food webs, widely distributed in the aquatic environment, and have been employed in water and sediment quality assessments. They respond positively and are sensible to chemical compounds as pollution indicator by different classes of compounds. For example, metals, pesticides, and endocrine disruptions. Here I show some species around the world that are studied from any kind of assays. Toxicity, mutagenicity, but the most important species around the world, okay? We have Pomacia canaliculata, distributed along all America, Paraguay, Uruguay, Brazil, United States. It's an important organism to these studies. Another from specifically in Africa and Asia, Bellamia. And we have another, this is very important here in the United States, Pophamophrygos antipodarum, and another for America, Bithynia tentaculata. We have another space for Europe and Siberia, Lithoglyphus naticoides. We have a north and Europe, another species, Physa acuta. It's very important species for this kind of studies. Valvatia piscinalis from North America, Europe, India, and Tibet. We have another Chilina gibosa, specifically from Argentina and Chile. Hedix auricularia from Europe and Asia. And Liminea stagnalis, a very important organism today. I explained better this species, and specifically more to Europe, but in Asia we can saw this species too. Here you can see the egg masses, and the adult and the egg masses both are used in ecotoxicological assays. And this another species, Planorbius corneus, to Europe and Asia, and Bionfalaria glabrata to South America. I choose this species specifically because I review all the species most usually in the toxicology and environmental analysis. And look at all these species, you can see some important assays used for ecotoxicology assays. I organize some assays that are established with some species, and I use my species to explain these assays, okay? Not only for my organism, but I work with this organism in Brazil. But some assays are the same, in another countries. So here we use acute and chronic toxicity for adults and embryos assays, and we analyze death and malformation embryos. This is our most usually organism to this kind of study. And you can see all the stages on this organism. For this, I put this information here. See. Whoops. See. You can see all the stages of the organisms. So you can analyze the adult effect and the embryo effect. Is the focus of the OECD and in the your recommend use embryo larval development. So you try to use this organism and Liminea in all parts of the world to do this kind of studies. Can you see all the developmental stage of the mollusk? And we established another assay specifically to Bionfalaria glabrata. Use male, here, male, and female, not female, but albinism as used as genetic marker. This assay is firstly used with rodents. This assay is usually to see malformed embryos after exposed the animal. So you can see all the progeny affected about many times after exposure. This kind of tests only to do with my organism, not with Liminea or another animal. So it's an important test to verify mutagenic compounds. Okay. We can see the progeny of these snails, and the analysis of little effects in the heterozygous offspring indicates the induction of mutation of germ cells of the exposed wild-type snails. These tests are standardized, but not much used yet. Another assay is very important. We saw about humans and rodents and comet assay. We established this test with Bionfalaria glabrata too, and now is very, very important test in laboratory, okay, for environmental analysis. And I need to talk something about this species and the importance of these tests. Toxicity and mutagenicity, specifically with the focus in embryo larval effects. Nowadays, we use most of tests with algae, microcrustacean, frog, fish, and we put this organism with potential to these tests because it's very easily to maintain, very easily to reproduce, is an important organism to make the environmental analysis. And in the first time in Europe, the OECD organized a norm to these animals. Liminae stagnalis is the first animal in the world that have a norm like algae, microcrustacean, and another used in laboratory as Daniel Herriot and other species. So we saw in the literature and the importance of improvement or established a new organisms specifically for country, for biota local, and you can use native or exotic. But the most important is improve the native organisms. For this, in Europe, use Liminae stagnalis, and they join 14 laboratories working together and have more information about this organism, and now will be the first norm about these mollusks. And from gastropods in America, we have some perspectives. Here in USA, much is forces in Potamophyrgus antipodarum. There is a lot of studies with this organism. Another possibilities is from Latin America, some countries in Latin America, Pomacia canaliculata, and some countries Chilina gibosa, because these organisms have more works, more information about biology, about reproduction. You need a lot of information about these organisms to establish a new test. And we now in Brazil, we work a lot with Bionfilaria glabrata, more than 30 years, with a lot of information. These organisms is important for us because they transmitted the schistosomiasis, so endemic disease, an important organism, and we have a lot of information about your biology, about your lifestyle. So in this moment, we work together with another groups in Brazil, Institute Butantan, where I'm involved my work, and for another researcher in south and north from Brazil to join this information and try to make the how can I say, make it this finally to be established, because we have a lot of information, we standardize the tests, but we need to join researchers and join the information for to put another organisms to the regulatory agencies. So we hope that the new steps, and we can discuss about the use mollusks in environmental analysis with algae, microcrustacean, and the fish, because we have a lot of these organisms all over the world. It's the second most important phylum in the world. So we try to put them in environmental analysis. Okay. I thank you for the people here and specifically the organizing committee that invite me to bring these informations here. I put all together, but if you wanted to talk about it, I'm here to talk with you. Thank you.
Questions? No questions.
No questions, or a short question.
Are there any questions? Yes.
Which is the type of celomocyte do you use?
Hemocytes? Release the hemolymph with hemocytes. You understand?
Yes, I know. Okay.
But it's very easy to collect it, and you don't.
You don't need to extrude the hemocytes out of the body.
You don't need to kill the animal.
No, of course, I'm not telling you
to stimulate
that.
the foot and release the hemolymph. You can use the animal
Together with the
one time,
mucus.
2 times, 3 times, and don't die the animal.
Yeah.
Don't.
Thank you.
Not necessary to kill the animal.
Thank you very much. Now I'm going to introduce you to my co-chair. Her name is Dr. Alenka Franco. She is an associate professor and an MD, is a specialist in occupational medicine at the Clinical Institute of Occupational Medicine in Ljubljana, Slovenia. Her research and teachings focus on several themes: occupational and environmental toxicology, molecular epidemiology, genetics, and gene-environment interactions, and occupational medicine. She carries out work in these areas nationally and internationally, has been a speaker at many national and international conferences. She's going to talk on the gene-environment interactions and asbestosis. Yes, there it is. Thank you very much, Alenka.
Thank you. So it has become increasingly obvious that both environmental and genetic factors may influence the development of many diseases. And primary candidates for gene-environment interaction studies have been mostly enzymes involved in the detoxification or in the metabolism of foreign chemical substances. According to the model of causation, exposure to different chemicals, time of exposure, genetic factors, and possibly also some unknown causes may have a crucial role in the occurrence of diseases. All these factors may act independently or more frequently interact or modify each other. So the gene-environment interactions will be presented on the example of our study into asbestosis, which is one of the most frequent asbestos-related diseases. And although the causal relationship between asbestos exposure and asbestosis has been well known, relatively little has been known about the genetic factors that may influence the development of this disease. In the pathogenesis of asbestosis, as you well know in the pathogenesis of many other diseases, the reactive oxygen and nitric species are involved. Among the latter, the most common is nitric oxide, produced by inducible nitric oxide synthase. But luckily, human tissue contains specific enzyme systems to detoxify reactive oxygen and nitric species. And superoxide dismutases and catalase, as you well know, present a part of the primary defense system against reactive oxygen species. Another important enzymes involved in the detoxification of reactive oxygen and nitric species are glutathione S transferases such as GSTM1, GSTT1, and GSTP1, which catalyze the conjugation reaction of reduced glutathione to different electrophiles. And the genes coding for all these enzymes are known to be polymorphic. And to explain genetic polymorphisms, at least some of them, to you, so this is a part of DNA, actually nucleotides, and this is a part of enzymes, actually amino acids. First, some examples of singular nucleotide polymorphisms. The most common polymorphism of the manganese SOD gene is a cytosine to thymine transition, which results in alanine to valine amino acid change. And in the extracellular SOD gene, a cytosine to guanine substitution causes an arginine to glycine amino acid change. Considering manganese SOD, so 3 genotypes are possible, and the same holds true for extracellular SOD gene. All these genotypes resides, of course, in different enzyme activity. Also, one of the most common polymorphisms of catalase gene is a cytosine to thymine substitution. In the GSTP1 gene, an adenine to guanine transition at exon 5 results in isoleucine to valine amino acid change, and at exon 6, a cytosine to thymine substitution causes an alanine to valine amino acid substitution. Regarding only exon 5, 3 genotypes are possible. Each of us has one of these 3 genotypes. And regarding exon 6, again, 3 genotypes are possible. But based on the presence of the polymorphisms in both codons, GSTP1 genotypes are known to be classified into groups with presumed high, intermediate, or low enzyme activity. GSTM1 and GSTT1 gene are known to exhibit another type. This is null or deletion polymorphism, which results in the absence of the active enzymes. So there are people with or without an active enzymes among us. And finally, one of the most common polymorphisms of the inducible nitric oxide synthase gene is a pentanucleotide repeat, resulting in short, long alleles, different genotypes, and again, different enzyme activity. So the basic aim of our study was to investigate the influence of gene-gene and gene-environment interactions on the risk of developing of asbestosis. The nested case control study included 262 patients or cases with asbestosis and 265 controls. These were subjects without any asbestos-related disease. Data on smoking were collected for all subjects during interview using a standardized questionnaire. All the subjects included in the study were, in the past, of course, employed in the asbestos cement manufacturing plant of Salonit Anhovo in Slovenia, and in the past occupationally exposed to asbestos. Asbestos was namely banned by law in Slovenia in 1996, but considering that it is a very resistant, it is still present in all our in the environment, not only in Slovenia, but unfortunately all over the world. It's a still big problem, especially because of the latency period, maybe up to 60 years or even more, 60 years, yes, according to our new data. So the data on cumulative asbestos exposure is expressed in fibers per cubic centimeters years were available for all the subjects from our previous study. The diagnosis of asbestosis or no asbestos-related disease was confirmed by experts of the State Board for Recognition of Occupational Asbestos Diseases, and it was based on the Helsinki criteria and on the American Thoracic Society recommendations. For genotyping, capillary blood samples from fingertips from all cases and controls were collected on the FDR mini cards, and different PCR-based methods were used for genotyping. In statistical methods, of course, logistic regression analysis was used to assess the association between asbestosis, genotypes, cumulative asbestos exposure, and smoking, and also age and other possible modifiers or confounders. And now the results. As expected, an important association was found between asbestosis and and cumulative asbestos exposure. However, no association was observed for smoking. Analyzing the association between asbestosis and different genotypes, as you can see, an important association was observed for manganese SOD alanine genotype. A slightly elevated risk was observed also for the extracellular SOD and catalyzed genotypes, but as you can see, the results were not statistically significant. Regarding GSTs, no association was observed for the GST2I null genotypes, while the presence of the GST2I null genotype suggested a protective effect, not an increased risk. On the other hand, GSTP1 isoleucine genotype increased the risk of developing this disease. And lastly, a slightly elevated risk was observed also for inducible nitric oxide synthase, but the result was borderline significant. So as you can notice, the genes may have no effect. They may decrease or increase the risk of developing diseases. However, genes, as for instance manganese SOD and catalyze, usually do not act independently, but may interact. So next interactions between different genotypes were tested first by simple categorical models based on stratification. And as you can notice, an increased risk of asbestosis was observed only among those subjects with combined manganese SOD alanine valine and valine genotype, who also had catalase TT genotype. This result suggested interaction, which was, of course, further confirmed by logistic regression analysis using dummy variables. So an important finding of the current study shows that the association between manganese SOD genotypes and the risk of asbestosis is strongly modified by catalase polymorphism. And whenever we find an interaction, we always have to prove that it is also biologically plausible. And as manganese SOD and catalase, as you well know, constitute a part of the primary defense system against reactive oxygen species, and as you can see, act in the same direction. This interaction could be considered as logical and biologically plausible. Similarly, an association between asbestosis and inducible nitric oxide synthase was also modified by catalase, a genotype, and also in this case the modifying effect was confirmed by logistic regression analysis. And considering that reactive oxygen species and nitric oxide have been proposed to cooperate in causing both cytotoxic and mutagenic effect of asbestos fibers, this interaction could also be biologically plausible. Next, I will present an example of interaction between smoking as important environmental factor, or let us say bad habit, and genotype, in our case GSTN1. In this case, GSTN1 polymorphism has been shown to modify the association between smoking and asbestosis, where increased risk of this disease was noticed only among those ever smokers who had a GSTN1 deletion polymorphism and consequently didn't have an active enzyme. Again, the interaction was confirmed by logistic regression analysis. So this study demonstrates a strong interaction between GSTN1 polymorphism and smoking, despite the fact there was no independent association between either GSTN1 polymorphism or smoking and asbestosis. The explanation could be that both asbestos and smoking increase the production of reactive oxygen species. And considering the role of GSTN1, this interaction is, of course, also biologically plausible. And last but not least, an example of interaction between exposure to chemicals, in our case, cumulative asbestos exposure, and genotypes, in our case, inducible nitric oxide synthase. The analysis showed that the association between cumulative asbestos exposure and asbestosis was strongly modified by inducible nitric oxide synthase genotype. As you can see, in both cases, there was increased risk of asbestosis, but this risk was much higher in this case compared to LL genotypes. Again, logistic regression analysis proved our results. So, and to conclude, the results of our study and also other studies suggest that in addition to environmental or occupational exposure to chemicals, the genetic factors, the gene-gene and gene-environment interactions may also have an important influence on the development of diseases and should be further investigated. And we are just working on the whole genome analysis right now. So these findings may namely serve as a basis for the development of new methods for an earlier diagnosis of diseases and also to identify new targets for a more effective treatment. Additionally, they could add to our understanding of pathogenesis of diseases and enable their prevention. In this way, they could significantly contribute to the improvement of the a quality of life, as well as to prolonging lifespan and aging. So thank you for your kind attention. And before that, I would like to stress that testing genetic factors should never be used for any kind of discrimination. This is very important, but just to thank help our patients. So thank you very much.
Quick questions?
Just as a point of clarification, when you genotype and you list the effects of a genotype, are all the genotypes homozygous or are they heterozygous?
Could you please
repeat? Yeah, are they homozygous? When you refer to GSTM1 null mutation, you're talking about just the presence of a single copy or homozygous? Okay.
Can I just ask, okay, do you still have asbestos? Sorry, do you still have people working with asbestos in your country?
Just removing the asbestos, the same as in UK. Asbestos is also banned in many EU countries, but still represent a huge problem, not only in Slovenia, also in UK, in other countries. We just remove asbestos, but the production is prohibited.
Thank you. Any further? So can we move on to the next speaker then, please? And this is Ying Pen.
to this. A lot of the slides refer to the same subject matter we can catch up on sometimes. So we know what sulfur mustard is. We are aware of these severe signs and symptoms. The mechanism behind it is DNA and protein alkylation, and as discussed before, this is leading to necrosis, apoptosis, and inflammation affecting skin, eye, respiratory tract, and in many cases also with severe systemic effects. Skin blistering is what's most prominent and most well known, and it's aggravated by a prolonged wound healing. I've talked about the Hackett cells beforehand, so I can now refer to the THP1 cells. It's also an immortalized cell line, and Hackett cells, they have been spontaneously immortalized under the influence of temperature and elevated calcium levels. In contrast, the THP1 cell line is a cancer cell line, so it had been derived from a one-year-old leukemia patient. But even so, these are cancer cells, leukemia cells. They have still retained a number of abilities of a physiological, of a healthy immune cell. So they are still capable of migration and most importantly of phagocytosis. In our model, we had two different approaches to either add these immune cells before or after the exposure to sulfur mustard. The exposure levels we used were similar to the previous study, 30 micromolar for subclinical, 100 to 300 micromolar for acutely toxic sulfur mustard challenge. The medium was not removed in any of the co-culture experiments because that would also remove parts of the immune cells and lead to irreproducible results. Tested compounds, when we tested for potential treatments of anti-inflammatory drugs, were added after 1 hour post-exposure incubation, and in some cases when we tested antioxidative substances, in particular glutathione and N-acetylcysteine, we had some experimental protocols when these compounds were added simultaneously with the sulfur mustard challenge. You've seen this picture before. After exposure and treatment, we conducted a 24-hour post-exposure incubation. We collected supernatant lysed cell layers and again did the assay of necrosis, apoptosis, and inflammation. And using this very nice automated pipetting system, we were come up with high precision data. So the intraday variation, the technical assay precision was about 2% to 3% relative standard deviation for necrosis data and 5 to 7% for all the other parameters under consideration. Biological intraday variations were approximately at 15% relative standard deviation. In the beginning, a few data still from monocultures. I had referred to silibinine before, but I would like to refer more closely to the addition of glutathione. When added simultaneously, they were really able to come up with a very pronounced reduction of necrosis, apoptosis, and inflammation. So these compounds, they are highly effective, at least against systemic, against circulating sulfur mustard, as long as this is present in the system or is again eluting from lipophilic deep compartments. Therapy using isoglutazine or N-acetylcysteine would be very effective to remove them more or less by a chemical scavenging reaction. Post-exposure treatment, I'd mentioned that before, was most successful with silibinine. So I can skip this slide. Why did we study the influence of immune cells? From a series of findings in literature, we had obtained the knowledge that immune cells were very much interacting with the pathology after sulfur mustard challenge. So immune cells lost their capability for migration and phagocytosis, but were also contributing to the inflammation. And our question was, could we possibly model this in our cell culture system? So in the first experiment, this is a set of our data when we first grew the co-culture model and then exposed the completed co-culture model to sulfur mustard. In the beginning, we used very different levels of immune cells. 2% would nicely correspond to the frequency at which immune cells, more specifically Langerhans cells, occur in the human skin. This is a strikingly constant ratio of 2%. In the beginning, we assessed that using this immortalized cells, this somewhat artificial model, maybe 2% might not be enough to come up with a significantly different response. So we step by step elevated this to 3, 4.5, 6 and even 10% of immune cells. But as you can see, there's no need to go to such a high level. It's really a matter of quality, not of quantity. As long as you have immune cells present, you have a very different and aggravated response to the sulfur mustard challenge. 2% of immune cells relating to the physiological ratio, that's enough to induce an aggravated cytotoxicity for sulfur mustard, and also to test compounds under more realistic conditions. There was, for the co-culture model, no need to increase the number of immune cells any further. Increasing the number of immune cells is, however, a very valid and interesting approach when we want to test the effect of healthy, that's to say unexposed to sulfur mustard, healthy immune cells migrating into the tissue after a severe sulfur mustard exposure, because in this case the number and the frequency of immune cells attracted to this focus of inflammation could in fact dramatically increase. So what we say is there is no aggravating mechanism anymore. If we add a large number of immune cells, they are even contributing to a mitigating mechanism, as you can see from this inflammation data. When you use 6 to 10% of immune cells, the inflammation is actually mitigated, probably due to the ability of the THP1 cells to to remove necrotic and apoptotic cell debris by phagocytosis. The conclusion from this is it's highly important to protect the immune cells, especially the circulating systemic immune cells. At the location of the poisoning itself, the immune cells will, no doubt about this, aggravate the symptoms because they are exposed to the sulfur mustard challenge as well. But it's really necessary to protect at least the systemic immune cells from the sulfur mustard insult so they are able to contribute to the mitigating and protective rather than to the aggravating effects. Well, we also used our established co-culture model to test some very well-known anti-inflammatory compounds. When I first set up this presentation, I purposefully blinded this, but now I'm able to tell you compound A is actually ibuprofen and compound B is diclofenac. I'm not going to present the glucocorticoid dexamethasone data because they were not really impressive. There was a slight but significant, in number terms, significant reduction of necrosis, but we ourselves questioned the clinical relevance of this rather weak finding. But what you can see is that under no circumstances, at least according to our data, you should use ibuprofen in case of sulfur mustard exposure because it's really, you can see here, the apoptotic response going up with I stick to the arrow, to the mouse arrow. If you increase the dose of ibuprofen, this is not going to help in any way. It's aggravating damaging the pro-apoptotic mechanism. And diclofenac, even so it's a compound from the very same class of non-steroidal, non-specific non-steroidal anti-inflammatory drugs, so non-specific COX inhibitors, it has really a protective effect. And with regard to, as you can see in the lower figure, and with regard to the anti-apoptotic and anti-inflammatory effects, this was the best compound we've tested so far. So maybe in combination with silibinine to rein in necrosis, diclofenac would actually contribute to the treatment. Now there's a slight COX-2 specificity, COX-2 inhibition specificity in diclofenac. This may contribute to the protective effects, but we would have to test this theory possibly by using some highly selective COX-2 inhibitors and to see if we could reproduce these encouraging findings. In summary, our co-culture systems are capable to rapidly assess novel compounds or for the treatment of sulfur mustard injury, or to test very well-known compounds for their efficacy and safety in case of sulfur mustard injury, because some of these effects can be highly surprising. We really didn't suspect the aggravating effect of ibuprofen when we launched the studies. The co-culture with immune cells is a better approximation of in vivo condition, and it's yielding more relevant and valid data. Silibinine currently provides the best anti-necrotic effects. TO compounds could have a role in post-exposure treatment, especially to eliminate the systemically to eliminate sulfur mustard after any systemic uptake. and ibuprofen should not be used, whereas the results of diclofenac are actually promising. I would also like to add that these findings are in line with some Israeli studies in literature which were done in animal studies, which also concluded that diclofenac was among the most promising anti-inflammatory compounds. So this ends my presentation, and now I'm happy to take any questions.
Thank you very much.
Is there a question? Yes, here's a question here.
I wanted to know from you what are the standard precautions you took and your colleagues during studying this hazardous chemical, which is sulfur mustard. Do you take a standard precaution to protect yourself and your colleagues during working with this hazardous chemical?
This is, of course, a highly relevant question, as safety is, as in every laboratory, our prime concern. Actually, as we work with very diluted concentrations of sulfur mustard, the day-to-day work in the laboratory, you don't have to don heavy protective equipment such as respirators or let alone any protective gear, protective suit. It has to be done in a regular laminar flow system, which is continuously monitored for its integrity, and of course, safety goggles and gloves. This is absolutely essential, but you could say that the safety measures are, when working with these dilutions, they are somehow equivalent to handling alkylating antineoplastic drugs.
Okay, thank you.
Thank you very much. Any further questions? Well, if so, further questions, if you could do them in the coffee break, please, because we have our new speaker now, our ex-speaker, who's already been introduced. He's now going to give his talk.
Okay.
Thank you.
Thank you. Good afternoon for chairs and everyone. Sorry for late because my PowerPoint miss in this computer. So my topic is autophagy elevated memory impairment caused by MAFI through inflammation suppress. I'm come from Shenyang University of Guangzhou, China. As you know, since 1806, MAFI has been widely used as potent analgesics, then induce many complications. Example, addiction, dependence, immunosuppressants, pituitary, memory impairments, etc. In 1917, scientists already found that chronic MAFI exposure could cause memory impairment. However, the chronic MAFI exposure will not cause deteriorating memory impairment like Alzheimer's disease. In 2010, the magazine Autophagy reported that chronic MAFI may cause autophagy in right hippocampus. In 2013, the Autophagy reported that MAFI can decrease a couple of number of mTDNA in hippocampus neurons in mice. The damage of mTDNA is the common cause of autophagy. So I think that is autophagy involved in toxicity or side effect of chronic MAFI explosion? as you know, the autophagy is a second type of programmed cell disease, different from apoptosis. It is an important safe defensive process for cells to keep service. It is related to tumor suppression, anti-infection, neurodegeneration, occlusion, stroke, cells, several, and starvation, etc. This picture shows the autophagic flux. When the autophagisome combine lysosome, then became autolysosome. The regulation of autophagy activated by the BACN1 and the ATGS, inhibited by BACE2 families. When autophagy occurs, the marker LC31 became LC32. The LC32 compared to LC31 increase, and the P62 degradation. So the question is, one, can MAFI really activate autophagy flux in the hippocampus in vivo? And two, is MAFI-induced memory impairment related to the autophagy? So I designed two experiments. The part one, to study the relationship between autophagy and MAFI-induced memory impairment. And the first step is choose the MAFI dose. I use the 5 microgram kg per day to 50 microgram kg per day. Then use the Western blot to detect the LC3 and the P62. This result shows when we use 15 microgram, the LC32 increase and the P62 and decrease. So it is here decrease. There's 15 increase. So the MAFI injected at the dose of 15 microgram for 7 days can activate autophagy in the hippocampus in say 50 cell mass. Then step 2, a dynamic change of MAFI-induced autophagy in hippocampus. This is the results. I found that 1 day after injection MAFI, the LC32 increased, the P62 decreased, the expression decreased. Oh, sorry. There's 1 day, 3 days, 7 days. 1 day, the LC3 increase, this is the expression, and P62 decrease. It is the MAFI. So the MAFI-induced autophagy activated from day 1 to day 7 in hippocampus with the strongest synchronous activation occur on day 1. So the step 3, autophagy activated regions, different regions in brain. I found that hippocampus, the BACN1 increase, the temporal level is increased. The hippocampus, the ATG5 is increased. Temporal level, no change. The basal XI, basal 2 in hippocampus, no change. This one, basal XI decrease. So the MAFI may induce autophagy in hippocampus in brain. Oh, this step 4, I divided 4 groups. this is a control group, this is a MAFI group, this is MAFI plus 3-MA and the 3-MA group. The 3-MA is a modulator for autophagy. When used alone, is a slightly active autophagy. When used a larger region, will be inhibit autophagy. This result show when you MAFI group, the LC3 increased, the P62 decreased, but MAFI plus 3-MA is recovered. But 3-MA used alone, there's a decrease, there's no change. This shows that this results. This transmission electron microscope, you can see this, the yellow arrow denoted the autophagy somes here. There's autolysosome. Tunisia can kind of apoptosis MAFI plus 3-MA is this group. This is the MAFI, this is 3-MA, this is control group. So the MAFI plus 3-MA, the apoptosis increase. So autophagy was activated in the neuron of hibiscus CA1 region in MAFI group and inhibited by 3-MA in MAFI plus 3-MA group. Result the more cell death in CA1 region of MAFI plus 3-MA groups. As 3-MA alone also induce a slight autophagy reaction that confirms the dual role of 3-MA in modulating autophagy. Then I just tested the Maris water method. This is the mouse trial from one side to a larger one side. So really the country group. But use MAFI, the many trial and cannot arrive there a larger side. This is a large group. This is 3-MA is a simple trial. This is a spatial occurrence. There's a distance travel to target. The MAFI and the MAFI plus the decrease. Also, this is a decrease. This current length and the border rate no change. Then, so I can get this conclusion that MAFI can induce memory impairment and autophagy inhibition in the hippocampal neurons of CA1 region, where aggregate this memory impairment by increased neuron death in CA1 region. In addition to the neuron itself, how will autophagy affect the microenvironment around the neuron in hippocampus? Post reported the autophagy induced by rapamycin can suppress the expression of INOS, is inducible nitric oxide synthesis, and AL6. This is inflammation factor. MAFI can induce immune suppression effect in vivo. This is reported in 2012. So I just do experiment party 2. This party 2 to study the relationship between MAFI-induced autophagy and inflammation within hippocampus. At first, purify the microgel. This is a purity of microgel. This APRIL-1 and the DAP1 as a mixture. Then also the microgel is also treated with the MAFI. When you use the 1 UM, the LC3 2 increase. There's a P62 decreased as with the dosage increase. The LC3 2 is increased, but the P62 in 1 UM and 10 units decrease, but 15 and 100 UM increase. So MAFI is also can activate autophagy in the micro gel. Is also divided 4 groups: control, MAFI, MAFI plus 3-MA, and 3-MA, 4 groups. Then I just detected the inflammatory factors, CD11, in MAFI decreased. This N-kappa B in the cytoplasm decrease, but in nucleoli increase. This demonstrated N-kappa B immigrated to the nucleoli. This is MAFI. This is MAFI. This is the MAFI plus 3-MA. 3-MA just inhibited the MAFI active autophagy. But another inflammatory factor, TNF-alpha, AL1 beta, AL6, and INOS increased. MAFI plus 3-MA. Then I think that MAFI inhibited the inflammation. So the MAFI induced autophagy, inhibited microgel activity, and suppress inflammation in hippocampus. Summary: 1. Chronic MAFI exposure activated the autophagy mainly in hippocampus. 2. MAFI induced autophagy reduce neural death in CA1 region of hippocampus, resulting in partial protection against the memory impairment caused by MAFI. 3. MAFI induced autophagy in hippocampal microgel suppress inflammation through NF-kappa B pathway, which is in turn confer protection for neurons. Thank you for your attention.
Two questions there.
Thank you for the presentation, but you used swimming maze. You used swimming maze for the memory. As I know, swimming maze is used for screening of antidepressant. So how you score memory in the swimming maze?
What memory?
This is in step 4.
Yeah, this is the. What is that?
Yeah. Yes.
Yes. This Meredith water maze just tested the memory ability.
Memory can use Y maze, not swimming maze.
Oh.
As I see now in your result that morphine has antidepressant-like effect because it struggle more with the treatment. You see? Compared to the control.
This is control, yeah. This control is a very simple. This trial very short time from one side to another side, but use the morphine. It's very difficult to one side, another side. Just around this.
Yeah, that's what I mean.
Yes.
He's struggling more. It means that he didn't enter the hopelessness stage with the treatment compared to the control. So morphine, by this result, I can say that
morphine 1 day,
has,
2 days, 3 days, 4 days.
has antidepressant-like effect. That's what I mean. I want to ask how you score memory using swimming maze. That's it.
Just talk with you after meeting. Okay.
Question behind you.
Not so much a question, but a general comment. I'm not really an expert on this, but it seems fully plausible to me, as you had outlined in your presentations, that a maze is a capable and a feasible approach to test memory functions. And as we can see, if this was solely limited to antidepressant effect, then, well, the score should improve under morphine, and it does not. It is slightly impaired by morphine and more severely impaired under the 3MA treatment. So it's your approach seems very plausible to me.
What do you mean? Yes. 东兵，我都快死紧，我是，我都快死。
Possibly, maybe the mice developed depression, because I did some neuroscience study. If the mice is depressed, they will stop swimming and just hanging there. So this one definitely can support this experiment can support his conclusion. It's increased the memory.
Yes. Yeah. I agree. Your idea. I agree your idea. Yeah.
Thank you very much.
Oh.
So we think, how do you think about the coffee break and post the presentation and afterwards to continue with last two speeches? It depends on.
Is that all right?
Is that all right?
But the last two.
Actually, they are. Yes, they are true.
Yeah, so 2 more speeches when we come back after posters and coffee, all right?
Or would you prefer to continue with the session? Continue with the session. Okay.
Is that all right for the coffee people?
For the coffee people, okay.
Right.
Yes, okay.
Okay, I'm sure they want to finish.
So our next speaker is Dr. Shaolin Wang, who will present environmental exposure to BDE47 is associated with the risk of diabetes. It is a community
Thanks for
chair's introduction. My title is a little long. I'm Sony Wang from School of Public Health, Nanjing Medical University, China. My talk is environmental exposure to classic chemicals, BDE47, is associated with risk of diabetes. This study to two parts. One part is community-based case control studies and animal experiment. PBDE is a class bromelate famous restaurant. It's used widely as additives in polymer for the electrical or electrical goods and also many areas. These chemicals have been detected in air, soils, waters, and bio range from the invasive to humans, and the levels has increased in the past 30 years. In 2009, PBDE was classed as a POPs by the Stockholm Conventions. Yeah, it's two very popular and classic pollutants. These chemicals, this is a Chinese map. The PBDE is highest concentrations in developed coastal cities. You know, it's a very too high. And in fact, child and adults to expose to PBDEs by different pathways. Infant exposed to PBDEs by milk. Children and adults are exposed to through the indoors dust. The BDE47 is a dominant PBDE congeners in humans, wildlife, and the environment. So this is about 7 BDE congeners, and we can the yellows is 47. So we can find the fish, infant food, and dietary supplements, animals or plant oils, eggs, meat, and milk is 2 highest. So in these studies, we selected these congeners to investigate the relationship between the BDE explosions, I mean the environmental explosions, and chronic disease. BDE47 has reported to induce many damage, including development, reproductive, neurological toxins, and disruptors of the multiple endocrine systems, including reproductive toxicology, toxicities. Another topic is type 2 diabetes. Diabetes is a popular chronic disease, including in China, especially in China. Now, this is 2 years, 3 years, 95, 2000, 2010. There is a world with the rate is increased, especially in China, in Asia, it's the same. In China, in the current several years, with the rapid economic growth and associated industrializations, And a lifestyle change, huge lifestyle change, such as increase higher calories, higher fat, higher sugars, and higher structural diets, and decrease physical activities. So the type 2 diabetes is popular in China and increased fast. This is the data that shows in 1994, the prevalence was 2.5%, but the studies between 2007 to 2008 show that the rate reached 9.7%, and pre-diabetes reached 15.5%. Males might be a little higher than females. And recent data shows that in China, the prevalence rate of the diabetes was estimated to be 11.6% in China. So China is a huge population. So with a high rate of pregnancies, so I think there is a much amount of the type 2 diabetes in China. It's a very popular problem in China. Also, like the other chronic disease, type 2 diabetes is affected by the genetics and environmental factors. And many studies from the GWAS studies just show that a genetic variant just might explain only 10% of the phenotypes variabilities. So environmental factors may be the main factors to induce the type 2 diabetes. This is the figures to show the environment induced the diabetes. So we can find this is the pancreas islet. And environmental effects can affect this site and induce and through the many cellular factors to induce the isolated cells apoptosis and this induced diabetes and also other phenotypes. In the other hand, the lifestyles after changes fast in China, or like for example, oversize. Oversize can be source many factors to induce insulin resistance. This is this can directly induce the diabetes. So we also can other the process. So high fat diets is a critical lifestyle diets related to the diabetes. This is data from animal experiment to find the high fat diets to induce the fat livers, insulin resistance, pancreas acid proliferations, inhibitions, and testosterone synthesis. PPD was reported to related to the glycolysis metabolism disorders, including diabetes, but the data or evidence for this relationship is snide. So, and the mechanism is remain unclear. So, in this study, based on our previous research, we conduct the two parts of the study to investigate the relationship between environmental exposure to BD47 and diabetes from human studies and an animal experiment. In human studies, we conducted two independent community-based case control studies from two cross-section studies. And in study one, we exploited the 245 pairs of case controls, and this result was validated further using an independent case control studies in study 2. Also, we conducted two animals experiments to confirm the human study result, and try to find the environmental exposures to BD47 and type 2 diabetes. The part one is associations of environmental exposure to BD47 with the risk of the human diabetes. These studies is human studies. And these studies were conducted in province, Jiangsu province. Jiangsu province is a very developed province in China. These are yellow seas, the coastal seas. So we conducted study 1 and study 2 in this province. These two areas is a distance 350 kilometers. This is independent too. So the studies, the study ones, I mean the core study ones. Core study ones and core study means cross-section studies. Yeah, cross-section studies. We enrolled about 2,700 subjects and another 600 and 200 subjects in the study 1 and study 2. And also the study 1 paired case control studies just from crude study 1 and crude study 2 perspective. And also the subject was underwent health examinations and face-to-face questionnaires. And for the diabetes diagnosis, the standard is if there was previous diagnosis in the hospitals, it just means authorized. And also inside examinations, there's fast plasma glucose is more than 7 millimoles, at least twice in different periods during studies. So the study 1 and study 2 just conducted from the crude study 1 and crude study 2. These paired age and 6 matched. For cross-section studies 1 and study 2, we just investigate the prevalence rate of the diabetes in the subjects. And the males and the females, in these two studies, the males or the females is similar, just a total of 9%. This result is similar to our previous report from China. Males might be a little higher than females. And For the to invalidate the expressions of PD47, we establish we detect these chemicals in the humans' urines. We detect the 7 congeners of PDEs, including the PD47s. This is the quality controls. And we find about 4 congeners of PDEs. The detection rate is more than 50%, like 23s, 47s, 144s, and 183s. Most of the human studies, we detect the distributions or types of characteristics of in case and controls. This is the main characteristics of, including family history or diabetes, hypertension, and others index offside. So we find these controls and case is very significant. All the index in the case is more than control. Study 1 and study 2 is the same. This data just shows the PPDE's congeners concentrations in this study 1 and study 2 of the case and controls. We just find the concentrations, the PD47s, either the detection rate or concentrations is significant. The case is most higher than control. Study 1 is study 2 is similar. So the study 2 is validated the study 1 result, but others congeners we didn't find this relationship, this result. Also, we calculated adjusted the ORS, odor ratios, and 95% CIs of the diabetes by the PD47s. So if we cut the values as not detectors and detectors, we cut detectors is the 2 parts, 1 tertiles and 2 first tiles. So the concentration is highest. So we can find in study 1 the ORS is in the now concentrations is 1.13, and the highest concentration is 2.1. And also, this result was found in the study 2. It's also the same. So we think environmental exposure to PD47 is maybe associated with type 2 diabetes. And
can I just say you have
okay,
3 more minutes?
Oh, okay, 2 minutes. Okay, good. Okay. To confirm the result, we just conducted 2 animal experiments. The first experiment was just to use a rat to expose to these chemicals, and we designed the 3 groups, and with just and for the 8 weeks, we didn't find the generous toxicities. And we also detect the PD47 in the plasma and the liver, so we can find it with giving the dosage. So the insult in plasma and liver's concentration is increased. Also, to find the antioxidant index is changed in those dependencies, and we find the glucose faster glucose is increased with dose, and the insulin is decreased with the dose. We can also find the livers and pancreas, the morphologies, which we didn't find the significant change in the pancreas, and a little change in the livers. And then we conduct the gene marker arrays. We just and then we teach the bioinformatics, and we find about 10 pathways in these pathways. The diabetes pathways is first, and some related genes was found. Also, they use RT-PCRs to confirm the genes, and the result is the same. And also, we do the bioinformatics to conduct the interactions of the genes. We can find the related diabetes genes, like the 3 is evolved. And also, we can do the co-expression network in this area. Also, to find the support the data. And to further confirm the result, okay. Just 1 minute.
Sorry.
Okay. Oh, sure. Just we conduct another experiment, and also we get the same result. Okay. So this is a primary data. So we need to do more experiment to confirm our result. Thanks. Sorry.
Thank you very much, but I'm afraid there is no room for questions.
But
so
we'll be having coffee after the next talk.
Okay. Maybe we can talk after meetings.
It was like a very good study, I must say.
That's okay.
So, thank you very much for an excellent presentation. And now our last speaker, Dr. Somat Paul from Indian Institute of Chemical Biology.
Wait a minute.
This is
now.
glutamate-induced cerebral toxicity, the possible role of nitric oxide in adult albino rats.
Okay,
Please.
thank you. From the land of 700 years of history, of ancient history, from Arab Republic of Egypt and from a small country governorate, which presents a great gift to the whole world, which is the new Suez Canal. I would like to present myself. I'm Dr. Heba Youssef, Assistant Professor of Forensic Medicine and Clinical Toxicology in Faculty of Medicine, Port Said University. And it is an honor to participate with all of you in the 4th Global Summit on Toxicology in Philadelphia, USA. The topic will be monosodium glutamate induced cerebellar toxicity and possible role of nitric oxide. So, as an introduction, monosodium glutamate is a salt form of L-glutamic acid and is commonly used as a flavor enhancer worldwide, especially in Chinese food. And several studies indicated that monosodium glutamate, with its glutamate excitatory characteristic, can disrupt the metabolism and the function of various organs, such as liver, thymus, ovaries, spleen, kidneys, and many parts of the brain, especially the cerebellum. Nitric oxide is produced by nitric oxide synthase enzyme, and it characterized by 3 distinct isoforms, which are endothelial nitric oxide synthase, neuronal nitric oxide synthase, and inducible nitric oxide synthase. And we enjoyed many presentations that talk about INOS. Nitric oxide is known to be responsible for the organization of many biological events in mammalian bodies as a second messenger and as well as a neural messenger. The nitric oxide has the highest level of expression. The neuronal nitric oxide synthase has the highest level of expression and of its protein content in the cerebellum, and is comparatively higher than other brain regions. Free nitric oxide is highly reactive and unstable. Therefore, it is difficult to measure nitric oxide. But fortunately, nitric oxide reacts with oxygen to form nitrite and nitrate, and so we can measure nitrite and nitrate as a measurable form of nitric oxide levels. So the aim of this study is to investigate the effect of non-selective inhibition of nitric oxide synthase enzyme isoform on the cerebellar structure and function in normal rats and in rats with monosodium glutamate-induced cerebellar toxicity. Material and methods. First, the animals, we used the 32 male adult albino Sprague Dowley rats of body weight ranging from 180 to 200 grams, purchased from the Center of Experimental Animal Faculty of Medicine, Zagazig University, and the study was performed in accordance with the guidance of the ethical care of animal and adopted by NIH Bethesda in 1985. All rats were left to acclimatize for 1 year prior to the study and are housed in plastic cages, maintain the control at a temperature, room temperature from 22 to 24 degrees Celsius, and with 12-hour diurnal change with free access to water and diet. The rats are grouped into 4 equal groups of rats matched by weight. Each group contains 8 rats, and the first group is control group, the second group is LNM treated group, third group is monosodium glutamate treated group, and the fourth group is LNM plus monosodium glutamate treated group. And I will explain in detail these groups. In group 2 and group 4, which are LNM treated group, LNM is abbreviation of L-nitro aspartate methyl ester, and this is a broad spectrum inhibitory pharmacological agent used to inhibit the nitric oxide synthase enzyme. So we use LNM by intraperitoneal injection at a dose of 100 milligram per kilogram dissolved in saline for group 2, and we administrate it 1 hour before monosodium glutamate in group 4. For monosodium glutamate treatment groups, which represent group 3 and group 4, we used here animals subjected to monosodium glutamate in a dose of 6 milligram per kilogram by IV injection. for 10 days, and LNM injected before monosodium glutamate in group 4, as we mentioned before. The second methodology we will use is Rota-Rot test. Rota-Rot test is a test used to assess the balance and the motor coordination, which is the function of the cerebellum in rats. And rats are placed in the rod, which is about 10 centimeter in long, and it is 4 centimeter in width or in diameter. And then the rota rod is automatically adjusted to round in 20 rounds per minute. And each rat were left on the rota rod at a time to spend from putting the rat until it fall down on the base of the instrument. This latency period from placing the rat until it fell down, this time, latency time is measured in seconds. It is about 2 minutes in normal rats, which have normal cerebellar or motor coordination function. Followed by measurement of cerebellar nitrite concentration. After motor coordination and balance assessment, rats were anesthetized, decapitated, brains are quickly removed and washed with acetylsaline, and cerebellar hemisphere were taken and divided into 2 parts. One part are used for histological and immunohistochemical assay and evaluation, while the other half were kept in -80 degrees Celsius for subsequent homogenization and measurement of concentration of cerebellar nitrite. Then followed by histological and immunohistochemical examination. And in this study, we used 4 types of dyes. First, hematoxylin and eosine, followed by Mallory phosphate tangustic acid hematoxylin, cressyl phosphate violet, and immunohistochemical stain using polyclonal antibody to Caspase-3 for detection of apoptosis. Also, quantitative assessment were done for the histological and immunohistochemical changes of the cerebellum in form of measurement of the mean dendritic length. The optical density was done for Nessel granules and Caspase-3 expression. The results. First, for the Rota-Rot test, it appears that the studied groups, the LNM treated group, the monosodium glutamate treated group, and both LNM and monosodium glutamate treated group show significant decrease in the cerebellar function when controlled with the compared to the control group. Also, cerebellar nitrite level from this table, we can find that there is a significant, there is a significant decrease in the level of nitrite level in group 2, 3, 4, the studied groups when comparison with the control group. There is a significant decrease. As regard to the histopathological results, this is a photomicrograph of a control group showing section in the cerebellar cortex, and it show here there is a normal stellate cell, normal basket cells, and this is the molecular layer. Also in the granular cells there is a pyriform Purkinje cells and granular cells. This is normal histopathological change. While in LNM treated group, there is appearance of distortion of the Purkinje cell. It becomes distorted, and there is increase in the intracellular vacuolation around it. While in monosodium glutamate treated, the Purkinje also distorted, but here in form of shrunken in the side of the Purkinje cells, and also it is surrounded with a clear space of intracellular vacuoles. In LNM plus monosodium glutamate treated groups, there is profound distortion of the Purkinje cells in form of deeply stained cytoplasm and deeply stained nucleus with vacuolated neutrophils in the molecular layer, not in the granular layer, and there is another Purkinje cells with karyolytic nucleus. For the optical density of Nessel granules, this is another histopathological result, but we use here cressyl fast violet in order to appear the Nessel granules within the Purkinje cells, and this is for the control normal group, so there is purple Nessel granules within the Purkinje cells. while in LNM treated group, these Nessel granules concentration decreased within the Purkinje cells. And also in LNM plus monosodium glutamate treated group, there is a decrease in Nessel granules in the pericrya of the Purkinje cells. When doing the percentage of optical density in the studied groups, there was a significant increase in the percentage of optical density of Nessel granules in the control group versus the studied groups. Another quantitative method is length of the Purkinje fiber dendrites, and this is the normal section in a control group showing bluish discoloration of myelinated nerve fibers in the white matter, while a photomicrograph of cerebellar cortex of LNM treated group showing shortening or decrease in the length of the Purkinje cell dendrites. In a cerebellar cortex of monosodium glutamate treated group, there is also decrease in the length of Purkinje cell dendrites. And in post LNM and monosodium glutamate treated, there is also decrease in the length of the Purkinje cell dendrites, but in doing statistical analysis, there was no significant difference in the mean of dendritic length of the different experimental group when compared with the control group. The third type of dye is Caspase-3 immunoreactive cells that reflect apoptosis, and this is a section in control group showing significant decrease in optical density of Caspase-3 immunoreactivity because Caspase-3 is only abundant and present in apoptotic cells. So the control group is not apoptotic, it is normal, so there is a significant decrease in optical density immunoreactive in control group. While in LNM treated group, there is brownish discoloration of Caspase-3 started to be appear and denoted by these white arrows, and this explains that there is apoptosis cascade will occur in LNM treated group. In group 3, which is monosodium glutamate treated group, also the white arrow here shows the brownish discoloration of apoptosis apparent by Caspase-3 immunoreactive cells. Also, again, appear in LNM plus monosodium glutamate group 4. When we putting this data together for the four groups, there is a significant increase in the optical density of Caspase-3 immunoreactive cells in the studied group versus the control group, since the probability is less than 0.05. In conclusion, inhibition of nitric oxide synthase enzyme disturbed cerebellar structure and function and potentiate monosodium glutamate induced cerebellar toxicity. Also, nitric oxide has a neuroprotective role in monosodium glutamate induced cerebellar toxicity. This is our home take message, and the recommendation further studies are required for better development of therapeutic strategy, and they are based on manipulation of nitric oxide synthase. And fortunately, I'll have a friend from Libya, from pharmacological department. I will have a collaboration work in order to try to find a drug for nitric oxide synthase inhibition and to be protective against the monosodium glutamate induced cerebellar toxicity. And last but not least, I'm honored to be with you in this respectable conference. And again, thank you.
Well, I can do it from here. I'm just going to ask you about monosodium glutamate in your country. Is it used a lot?
Yes, yes. Recently, there was many restaurants that represent food containing monosodium glutamate salts. And many young people used to go to this restaurant and eat this food.
It's just that in China, of course.
Yes, yeah. In Egypt, we have many restaurants that deliver Chinese food.
It's not used. It's not used to that extent.
No, yes, it is. It is. Yes, yes.